Principal, Department of Pharmaceutical Chemistry and PG Studies, Dr. Vittalrao Vikhe Patil Foundation College of Pharmacy, Virazghat. Sir, your rich academic experience, research contributions and membership have inspired generations of postgraduate students, and we are truly privileged to have you with us today. Thank you, sir. This program has been thoughtfully designed, keeping in mind the critical transition of Pharm students from coursework to research-driven learning. Research today is not merely a degree requirement. It is the foundation for innovation, evidence-based practice, and the academic excellence. The objective of today's program is threefold. First, to orient first-year Pharm students towards research thinking, project readiness, and scientific courtesy, also curiosity. Second, to equip second-year students with practical skills in thesis writing, ethical research conduct, and high-quality journal publication. And third is to bridge the gap between project execution and research impact, enabling our students to convert their hard work into meaningful scientific contributions. Through this expert session, we aim to strengthen students' understanding to research planning, data interpretation, scientific writing, plagiarism ethics, and publication strategies, which are essential not only for academic success but also for career in industry, research institutions, and higher education. Our institution strongly believe that the quality research creates confident professionals, and programs like this reflects our commitment to nurturing competence and ethical and globally competitive pharmacy graduates. I sincerely thank our management, principal, faculty members, and organizing team for their constant support and encouragement in making this program possible. I also appreciate the enthusiastic participation of our Pharm students, for this program has been specially curated. Once again, I warmly welcome all dignitaries and participants and wish everyone a highly encouraging, interactive, and intellectual rewarding academic experience. Thank you, everyone.
Objectives of the program. I now request our respected principal and convener, Dr. Shyam Pakka, to kindly address the gathering and share his valuable message.
The pattern of today's program, Sri Ramkishan Deshmukh Sir, Secretary Seva Shikshan Prasarak Mandal, Distinguished Resource Person, Dr. Ramesh Sawant Sir, Vice Principal and Senior Professor of College of Pharmacy, Virazghat, esteemed faculty members, dear students, distinguished invitees, a very good morning to one and all. It gives me immense pleasure to welcome you to this academic orientation and research skill enhancement program for Pharm students. On behalf of our institution, I extend a warm welcome and sincere thanks to our esteemed resource person for graciously accepting our invitation and sharing valuable expertise with our postgraduate students. The Indian Constitution, through its fundamental duties, calls upon every citizen to develop a scientific temper, humanism, and the spirit of inquiry and reform. As students and scholars of pharmaceutical sciences, these ideals are not merely constitutional values, but the very foundation of your academic and professional journey. This program is designed precisely to nurture these qualities by guiding you in ethical research practices, logical thinking, critical analysis, and responsible scientific communication through thesis writing and journal publication. I strongly urge our students to actively engage in today's session, learn from the expert insights being shared, ask meaningful questions, and cultivate curiosity beyond the syllabus. True research begins not in the laboratory alone, but in the mind that questions, observes, and seeks truth for the benefit of society and humanity. I take this opportunity to express my heartfelt gratitude to our Honorable President, Sri Vinaynath Paulbudhe Sahib, for their constant support, vision, and encouragement in promoting quality education and research, and for providing the necessary facilities and kind permission to organize such academically enriching programs. I also appreciate the sincere efforts of all the faculty members and organizing committee for successfully bringing this program together. Once again, I thank our resource person for contributing to the academic growth of our students and wish all participants a highly enlightening and productive learning experience. Thank you. Thanks for listening.
नमस्कार. आजच्या या कार्यक्रमाची प्रमुख अतिथी आणि ज्यांचं लेक्चर ऐकण्यासाठी आपण या ठिकाणी उपस्थित आहात असे याचे प्रमुख अतिथी डॉक्टर सावंत साहेब, उपप्राचार्य विखे पाटील फाउंडेशन, आणि ज्यांची धडपड सतत आपल्या विद्यार्थ्यांना काहीतरी देण्याची असते असे ॲक्टिव्ह प्राचार्य डॉक्टर पांगा सर, सर्व फॅकल्टी प्रमुख, प्राध्यापक, शिक्षकेतर कर्मचारी आणि ज्यांच्यासाठी हा कार्यक्रम आयोजित केला असे प्रामुख्याने फार्मचे विद्यार्थी आणि त्याचप्रमाणे डिप्लोमाचे विद्यार्थी. आपला सर्वांना डॉक्टरांचं योग्य असं मार्गदर्शन घेण्यासाठी आपण सर्व जमलो आहोत. त्यामुळे मी जास्त वेळ घेणार नाही, परंतु एकच सांगू इच्छितो की संस्था आपल्या पाठीमागं म्हणजे सर्व जे प्राध्यापकगण आहेत आणि त्यात आपले जे ॲक्टिव्ह सतत कुठल्या ना कुठल्या तरी कार्यक्रमामध्ये नवीन असे हर उन्हाळी डॉक्टर पांगा सर, की हे सतत मी आल्यापासून त्यांचं पाहतोय की विद्यार्थ्यांच्या सर्वांगीण विकासासाठी जे काही आवश्यक असेल ते करण्यामध्ये त्यांचं म्हणजे ते दररोज आठवड्याला एखादा जरी प्रोग्राम समोर मांडतात आणि मला आनंद याचा वाटतं की आमचे अध्यक्ष साहेब त्याला तत्वतः मान्यता देतात. कारण त्यांना खात्री आहे जो काही प्रोग्राम डॉक्टर पांगा सर किंवा त्याचे सर्व जे साथीदार प्राध्यापक करत आहेत त्याचा निश्चितच फायदा आपल्या सर्व विद्यार्थ्यांना होणार आहे. आणि म्हणूनच त्यांना तसा साहेबांनी फ्री हँड दिलेला आहे की तुम्ही या कार्यक्रमाचं आयोजन विद्यार्थ्यांच्या सर्वांगीण विकासासाठी अवश्य करा. त्यासाठी नेहमी संस्था तुमच्या पाठीशी असेल. मित्रांनो, आजचे जे प्रमुख अतिथी आहेत आपले म्हणजे खूप मोठे प्राध्यापक आहेत, डॉक्टरेट केलेले आहे. आणि मला असं वाटतं की आज फार्मचे जे पदव्युत्तरचे विद्यार्थी जे आहेत त्यांनी ह्या अशा मोठ्या व्यक्तिमत्वाचा निश्चितच आदर करून त्यांनी जे आचरणात आणलेलं आहे आणि जे एवढ्या मोठ्या पदावर पोहोचलेले आहेत, विखे पाटील फाउंडेशन सारख्या ठिकाणी आणि गेल्या 15 वर्षापासून 20 वर्षापासून ते कार्यरत आहेत, म्हणजे त्यांचं खूप मोठं योगदान आहे. आणि त्यांचे अनेक थेसिस वगैरे प्रसिद्ध झालेले आहेत. तर याचा लाभ आपण घ्यावा आणि आपल्या आयुष्यामध्येही आपण त्यांच्यासारखं किंवा नव्हे त्यांच्यापेक्षा मोठं होण्यासाठी प्रयत्न करावं. मला असं वाटतं की डॉक्टरांना निश्चितच त्याचा अभिमान वाटेल की माझ्यापेक्षा माझे विद्यार्थी मोठे झाले, हे खूप मोठे त्यांची गेनिंग असेल. विद्यार्थ्यांनो, हे तुमचं क्षेत्र संशोधनाचं आहे. त्यामध्ये तुम्ही कुठंच थांबायची आवश्यकता नाही, याला पूर्णविराम कुठंच नाही. आपण पूर्वीच्या काळात पाहिले की आपल्या जवळ 100 किलोमीटर असणारी सिरम इन्स्टिट्यूट सारख्या इन्स्टिट्यूटने जगाला लस दिली आणि जगातले माणसं अनेक लाखो व्यक्ती त्यामध्ये वाचवले. मला असं वाटतं की 100 किलोमीटर आपल्या जवळच आहे. त्यामुळं आपणही असंच काहीतरी संशोधन करा की जेणेकरून आपल्या देशाला आपला त्याचा खूप फायदा होईल. आणि तुमच्या निमित्ताने आम्हाला आनंद याचा वाटेल की आमच्या कॉलेजचे विद्यार्थी हे एवढ्या उच्च स्तरावर त्यांनी संशोधन केलं आणि एक कुठली तरी दिशा, कुठलंही निश्चित असं एखादं ध्येय घेतलं आणि ते पूर्णत्वाला नेलं. मी या निमित्ताने अशीच अपेक्षा करतो की तुमच्या आयुष्यामध्ये तुम्हाला खूप मोठ्या होण्याचा चान्स मिळो, खूप मोठं संशोधन करावं तुमच्या हातून व्हावं आणि तुमचं नाव ज्याप्रमाणे देशाचं नाव जसं उच्च कराल त्याचप्रमाणे आमच्या संस्थेचे नाव मोठं करा अशी आशा बाळगतो. आणि पुनश्च एकदा सर्वांना खूप खूप शुभेच्छा देतो. आगामी जे 2026 साल सुरू होणार आहे त्या वर्षासाठी आणि त्या शैक्षणिक वर्षामध्ये तुमच्यापैकी कित्येक जण चांगल्या मोठ्या कामगिरीला पुढे यावेत अशी आशा व्यक्त करतो. थांबतो. धन्यवाद.
Growth of institution since many years I am associated with, I think from inception rather, I can say that I am associated with this organization, and I can see the growth, the way it is growing, and I must appreciate the contribution of Principal Chavan Madam, Sujamsi Madam, Bhugarkar sir, Tade sir. Actually these are my students. And then Paulbudhe, Dr. Paulbudhe, to be Dr. Paulbudhe. So the kind of dedication, contribution they are giving towards the institution, that is the reason I was expecting Deshmukh sir to attend this, at least initial words, so that the message should reach to him. Rather, he will show this recording to the management, because the college grows only because of the team, right? If team is strong, if team is satisfied, then only organization grows. So I must appreciate the teamwork of all the faculty in this regard. One fine morning, your principal called me, "Sir, you have come for a whole day for session to the Pharm." I said, "Sir, whole day?" No, sir, whole day, sir. So, it gave me some pressure that, see, this is a era of 30-second reel. 30-second reel played with 2x. Right? So in this era, if I have to attend for a whole day, it's big pressure on me. How to handle? But see, whatever objectives stated by your faculty and principal, the heavy objective of this session, I will make it too light. Don't worry. It will be too light. No pressure to you. Right? That only you expect, na? No pressure. So, okay. See, no, keep it, no issues. Now, I hope all are Pharm students. So you are in Pharm now? Yes. You are in first year. Most of you are in second year. I can see the uniforms. The majority is in uniform, so I expect those who are in uniform are in second year. So you have covered, successfully completed first year in Pharm. Right? After completing 4 years of B.Pharm, then after completing 1 year of first year in Pharm, now you are in second year. Great achievement. Let me ask one question. How many of you have joined Pharm by choice? See, because I have to speak for a whole day, it is not my duty to speak only. You also have to speak. Right? It's very difficult. One side, it will not work. So, how many of you have joined Pharm by choice? Or just, I applied for GPAT, I got some score, so I applied to CAT cell, I got some allotment, I joined. Which is option A or B? Those who join Pharm by choice, those who have already decided that I am going to do Pharm after B.Pharm, so I will prepare for GPAT, since say from first year or second year or third year, whatever, prepare for GPAT, then qualify GPAT and enter in the Pharm. How many of you in this category? Please raise your hand proudly. It's not like, ooh, like don't give, right? And those who have joined Pharm because I got admission to the Pharm. How many of you? Including me? Okay. So, whatever may be the reason, whatever may be the motive, but now you are in Pharm. Right? So forget everything. How, why you came here, that is now no issue, right? So, when you are here, you should be like, You are a Pharm student. Right? So B.Pharm, during B.Pharm, what you did? Continuous assessment, exams, sessional, assignment, something, then university exam. Read something, write much. Read something from some book, write much. Don't know what to write, still distinction. How many of you are in distinction? In B.Pharm? Many must be in the distinction. How many of you in first class? You can raise your hand. If you have a problem, you can come forward. You both. First class? Most of the students are in first class. Right? Maybe 20-30% are in distinction. Right? 20%. So what is meaning of distinction, you know? Distinction, what is meaning? Why that word? How this word appears? Distinction. Something different. It is not common. Distinction is not common. Still, most of the common people are in distinction. Because it's university system. It is very, very difficult to fail in university exam. Very difficult. Right? You must agree with this. Right? Because you better know that what you have written in the exam and still how you got distinction in first class. You better know this. Right? So it is highly difficult to fail in university exam. This is what is B.Pharm course. Right? Correct? Now you are in M.Pharm. What is M.Pharm course? You know what is the difference between B.Pharm and M.Pharm? B.Pharm was completely theory based. You have to appear for examination. You will get the result and okay. But in M.Pharm, it is partly by paper, partly by research. Now what is research? Whatever you did in university exam is research? No. Why that curriculum has been designed? See, we are here to understand the curriculum, right? What is curriculum? How it is designed? Why this M.Pharm is partly by paper, partly by research and all this? See, in examination, whatever you write in a paper, come out of all, nothing is here. Because you have read to write only in the paper, not to carry, not to understand, just to write. Right? This is what is examination system is. But when you want to do some research, it is not for to write somewhere. It is to understand and then do something that can be beneficial to others. Do something new after reading. So here, reading is to not to write only, but to understand and do something. To make others to read it. This is what is research I can feel. Right? So, the curriculum of B.Pharm and M.Pharm is totally different. So you must understand how it differs and all. Now because you are in M.Pharm, it is based on research. You have to do some research. Means you are going to become a scientist. Correct? Now, how many of you want to be a scientist? How many of you? Honestly. Anyone? Any hand? I want. Anybody? Okay, how many of you want to be a politician? Politician, right, correct. You want to be a Amdar, Khasdar, or Chief Minister, or Prime Minister, or Governor? Amdar. Good, very good. How many of you want to be a IAS officer? Any officer in a government organization? Any officer, not IAS executive. Any officer. Okay. Okay. How many of you want to be a teacher, or professor, or a principal like that? Okay. Out of all this, who is the most respectable personality? Scientist or Who is most respectable? Who is most respectable? What do you feel? No, see, when PM is coming, we used to say, Honorable PM, Honorable Principal, Honorable Director, right? Then Honorable IS Officer, and then Newton. Do you see Honorable Newton? Right? Honorable Einstein, Honorable Einstein's theory, Honorable Newton's theory. Have you heard like this? So who is most respectable fellow? You hate Newton, you hate Einstein, because of them we have read and all this. Man. So, do you agree with me? The scientist is least respectable fellow because he is always called a singular. You see Einstein, Arrhenius, Bose, right? Agree? But generations to generations, we are remembering Einstein, we are remembering Newton. What about earlier prime ministers? Do you remember? Earlier IS officer, do you know? How many of you know? Right? So, this is a respect. Just saying honorable, it doesn't mean that the fellow is respected. Right? So, see, nobody is saying what you can tell Amar. Nobody is Amar. Right? Everybody has to die. There is one way to be Amar, you know. एक गोष्ट आहे आपण अमर राहत आहे लोकांच्या मेमरी मध्ये राहत that scientist can no any politician, you know. Okay, so think, or those who want to be a PM or whatever, or some Nagar Sevak. Now election is coming up next. Those who want to be, you can fill form, no issue. Ask for us to vote, we may vote, no issue, because any candidate can ask. Okay. So, to be a researcher, I am saying researcher, not scientist. Scientist is a far. We are beginners. We are just entering in this field. We are not scientists. Whatever introduction, very nice introduction she read, but it's dumb name. We are just beginners. What we did for the society, what society is getting from us. Have you seen any theory, Saman's theory? The day you will, if I don't get that Saman's theory, I am beginner also. Right? That is the Newton coming in, that is Arrhenius coming in, that is the Einstein coming in. So we are nowhere. Right? So what quality is required? It needs a patience and hardship. Do you have patience? How many of you have patience? Initially only I said, we used to watch 30-second reel with 2x speed. Means how much patience we have? See, we are not capable of listening others even for 30 seconds. 30 seconds. Then it will not help anymore. So, that quality of patience, consistency, passion, that is required to be a good scientist. Okay. Now, see, here I will tell you the curriculum design, who designed this curriculum, how the curriculum is reaching to the students and all. Do you know who designed this syllabus, where from syllabus comes? Anybody knows? We will start from basic things, right? So here I am here to show your 2-year journey, project your 2-year journey, right? So because once you have got admission to the Pharm, okay, now you will have some lecture starts and timetable, all this. So who gives this curriculum? We are following throughout India. The curriculum is designed by Pharmacy Council of India, the Central Council. Being a supreme authority, they expect that the curriculum designed by them should be implemented throughout the country, because they want like that. So the same curriculum is implemented throughout the country, every college. So that curriculum goes to the respective universities, because we are associated with Savitribai Phule Pune University. The PCI syllabus comes to Savitribai Phule Pune University. In Savitribai Phule Pune University, there are different board of studies like that. That members, the chairman, all this, they sit together. They approve the syllabus. They just approve, because they don't have right to modify, even though it is required. You can listen in between lines. Even though it is required, but because the council says that it has to be implemented as it is throughout the country. So that board of studies only just approved, yeah, yes, like, so it comes to the student. Then in this Pharm, it is of 2 years, 4 semesters, right? Theory, practical, seminar, journal club, research proposal, presentation, internal and external assessments. That we'll see next slide. Now, when you are in first year or first semester is like your routine Pharm. 4 subjects as per specialization. I feel here is pharmaceutical chemistry, pharmaceutics and pharmacology. So in first semester, 4 theory subjects, 1 practical. It is like your Pharm, right? Internal assessment test, practical, that's all. The most important thing every institute should do, it is for staff also, what I am going to say is, that you see that your guide development is done in the first semester itself. Though curriculum says that research project is undertaken in second year, that is 3rd and 4th semester, see that the guide development is done in the first year and first semester itself. See, I will tell you the reason. The role of guide is very important. How many of you understand Marathi? All. I will tell you one story. जंगलामध्ये एक लायनचं जस्ट जन्मलेलं पिल्लू असतं आणि त्याच्या बाजूला लायनेस मरून पडलेली असते, मेलेली असते. जस्ट जन्मलेलं लायनचं काय म्हणतो ते पिल्लू. मेंढ्यांचा कळप बाजूला चाललेला असतो. त्या मेंढ्यांच्या कळपातले जे कोणी लीडर आहे ते बघतात ते लायन छोटंसं पिल्लू एकच आहे, त्याची आई म्हणून पडली. त्यांना त्याचं काळजी वाटते, त्याला कोणी सांभाळणार. मग ते त्या लायनच्या पिल्लाला मेंढ्यांच्या कळपात घेऊन जातात. हे लहान जस्ट जन्मलेलं मेंढ्यांच्या कळपात वाढतं. कोण वाढते मेंढ्यांच्या कळपात? लायन. थोड्या दिवसांनी तो ग्रो होतो, मस्त लायन तयार होतो. पण तो सगळं खाणं पिणं सगळं जे काही ते मेंढ्यांसारखं चालतं. ओके, त्याला माहित नाही तो कोण आहे. त्याला वाटतं हे आपल्याकडेच आहे. मग एक लायन बघतो, अरे हा लायन त्या मेंढ्यांमध्ये काय करतोय, बघा इथं कोण लायन आहे का, कुठल्या मेंढ्या आहेत का, कुठला तरी लायन कुठेतरी मेंढ्यांमध्ये काय करतोय. मग तो सांगायचा प्रयत्न करतो. तो गाईड म्हणजे गाईड हा जो लायन सांगतोय त्या दुसऱ्याला, जो मेंढ्यांमध्ये राहणारा, अरे तू काय इथे करतोयस, तू लायन आहे, तू मेंढ्यांमध्ये काय करतोय. त्याला माहितीच नाही, त्याला कळतच हा काय. शेवटी तो लायन त्याला ओढत नेतो पाण्यापर्यंत. मग त्याचं प्रतिबिंब दाखवतो, तू कोण आहे. मग तो डरकाळी होतो. त्याला कळतं मी लायन आहे, मी मेंढी नाही. तर गाईडचा रोल आहे. म्हणून दॅट्स व्हाय व्हॉट आय सेड की in first year itself the guide development should be done so that guide can guide student, guide can realize that student what is its strength. So students should be interactive with the guide. That is why what I said, what I showed here, that guide allotment in the first semester. That's whatever PCI curriculum they have given, what I am expecting in between line, what I wanted to listen, that is this. Because one should, if you want to really or effectively implement all this, you see that you can make change, you can have your own autonomy, you can make some changes in the program and do like this. So do the guide allotment in the first semester, so that right from the first semester you can think of your research project which is going to be done in your second year. So that can be possible. Okay, next. Similarly, in semester 2, same thing, some 4 theory subject, 1 practical, assignment, internal test, seminar. Okay, now seminar, what is this seminar is? What is to be done in this seminar? Who decides seminar topic? What are the guidelines for selection of seminar topic? What are guidelines to conduct that seminars? I think those who are in second year, they must have given their seminars in first semester, second semester. I don't know what procedure you implemented, but see that in first and second semester, whatever seminars are there, these seminars or these topics should be selected from your curriculum subject. The topic selection and guidance to the seminar, the preparation, all this, that should be given by guide, because in the first semester only you are allotting the guide. So here again the interaction of student and guide starts for the seminars, right? So likewise in the first and second semester, seminars should be from your subject, your topic, and it should be guided by a person, a guide. That's why you have to do the guide element first. Then in first and second semester, you start defining your research topic, which you are going to perform in the second year, and you start your literature survey. See how much time we get for research in second year? 1 year. 1 year. How many working days in a year? 365 days, total days, working days I am saying. Maybe 190, including all vacations, leaves, everything and personal leaves, and all other special leaves. Some have some special leaves. Again, read in between lines. So including that all, how much effective working days you get? Do you feel that in that, say, 60 or 70 working days, you can do some research? Is it possible? No, not possible. Or otherwise, it is possible. Otherwise, right? How many times you can repeat the same experiment? How many times we can fail and repeat and improve? Do you have this 60 days or 90 days sufficient for this? Research doesn't happen like this. People, I can see the student, right? Sir, nahi aa raha, product nahi aa raha. Try. Sir, try किया नहीं आ रहा. कितने बार किया? सर एक बार किया. फिर दूसरी बार भी किया. एक बार किया, दूसरी बार भी वो क्या बोलते हैं? वो फेवर किया गाइड. दूसरी बार करने का. See, someone failed 10,000 times. Then he discovered that bulb. How many times? 10,000 times. Here, student gives favor to the guide that I repeated it once. See, got it? So this will not make any research. That's why I said patience is required. That's why I said whatever time we get in second year of Pharm, people are watch. You are in a hurry? No. You are not in a hurry. So you should have some patience, right? Then only it is possible. Otherwise, in 1 year or 60-90 days of time, the research or quality research is not possible. You have to begin early. That is the reason what I said is, in first semester, second semester, start your literature review, topic finalization, all this. Next. Now, in semester 3, there is research methodology and biostatistics. This subject is kept. Right? So everywhere the research methodology, biostatistics, validation, and everything is required. Right? So in semester 3, here you have to present your proposal for research. Right? This is called as a research envisaged. How your research will go. That is a plan of research. That plan of research should be presented in semester 3. Then the team, see, what I expect is whenever students are presenting their research or whenever they are giving their seminars, even first semester, second semester seminar, the faculty, those who can attend, every faculty should see that irrespective of their specialization. Even pharmacology faculty can attend pharmaceutical chemistry or pharmaceutics seminars. Because if faculty is sitting in front and even every student should be there, the hall should be full. There, the presenter can build his own confidence. Right? If sometimes what happens, what is the purpose of the seminars, all that? The overall growth of the student. If student is exposed for the faculty and a class full of students, class full of audience, then confidently he can deliver. Otherwise, in a restricted environment, in front of guide only, or only showing presentations on mobile, not even laptop, right? Right? Sir, presentation, mobile. Sir says okay. Then the student will not develop. The confidence will not develop. See, at our time, when I was doing Pharm, the whole class was full. The first year Pharm, second year Pharm, all students used to sit. Most of the faculty used to sit. You have to present for 30 minutes, 35 minutes, and 1 hour cross question. 1 hour. Everybody used to ask. Till you say sorry now. Okay. So that develops you. That gives you confidence. Right? If you feel that I am speaking confidently, the reason is that. Because in Pharm, everybody screwed us like anything else. The first seminar, we, after going to the room, we cried, literally cried. What is this? What they are doing? But the impact or output is this, you can see. So I expect that the faculty or student should try to inculcate this culture to develop a student. If you want to develop student, this thing should happen. Otherwise, गुड़ी अच्छा है, तुम खुश, हम खुश, डिग्री मिल रहा है, तो वैसे भी मिलेगा, एडमिशन लिया तो मिलेगा कि नहीं? B.Pharm मिला वैसे M.Pharm भी मिलेगा, एडमिशन लिया, right? So if you want to do something really, the hardship is required. Someone should be there to make you hard, make you to do some hardship. Okay. Then, so in semester 3, the proposal presentation, the topic approval like that, and the regular. See, one more thing, that once your topic is finalized, or once your guide is awarded, the student should be in regular contact with your guide. You should make some register, daily register. Write a date. I met this day, guide. What was discussion? You can write the abstract. What was discussed, and next time you first talk about earlier discussions, and then the next. Right? You have to maintain the daily diary with interaction with your guide. You know Gurukul culture earlier. क्या Gurukul culture? So विद्यार्थ्यांनी त्या गाईडला जाऊन राहायचं. नाही तर गुरुकडे जाऊन राहायचं. गाईड आहे गुरु. The Guru Ji whatever he follows daily routine, the student has to follow. Just follow. Just follow. And learn from him. And learn from him. That time this educational institutes, universities, nothing was there. Just go and get your Guru, in touch with your Guru, and follow him. Learn from him. Observe him. Are we doing this? What we are observing from your Guru? If you interact with your guide repetitively, something will be reaching to you from him. Something can be percolated towards you. If you are contactless, then you can imagine what is out. So no impact of your Guru or guide on you if you are contactless. See, that is the reason why I always insist that the guide allotment should be in the first year itself, first semester, so that you are in contact with your guide for at least 2 years, maximum period of time. So whatever you have to acquire from him, that can be acquired. Okay. So like that, semester 3rd, next. Semester 4, completion of research, interpretation of data. See, whatever research, everything that we are going to discuss in second presentation. Carol. See, so we will not talk on this, what is research, what is project, how to design project, then how to write dissertation, everything we are going to discuss in second session. So here just we are highlighting the points that are to be covered during this. So once in the second year or first semester you start your research, here what is expected in the fourth semester is you have to complete your project, complete in all respects. You have to complete your experimentation, you have to complete whatever data collection, everything. You finalize your whole research in the form of a thesis or dissertation, you can say. Then you have to present that initially in front of your internal committee. That is a fourth semester seminar, internal committee. They approve it, you write your thesis, complete your thesis, submit it, and then it can be presented in front of the external examiner. That is the whole process. Here one more thing is there, that is journal club. Have you heard about this journal club? And what it is, how to implement journal club? The purpose of journal club here is to make you to understand and read research papers which are published earlier someone else in journals. How citation is done? You know anybody? How, see any standard method. Whenever we cite any article, here we cite some first author's name, then we cite title of your research paper, then name of journal, then volume, issue, year, and page number. Those who are in second year now, anybody knows what is volume of that journal and what is issue, what is meaning of volume and issue? Anybody knows? Anybody from you, let me know what is meaning of that volume and issue in the journal when citation. It means that you have not seen the research paper. It indicates that you have not undergone this type of things. So it is required. Unless you don't know, unless you know this all, how you are doing your literature survey? How to search it? What is the meaning of literature survey? How many of you know literature survey? Student used to come, sir, I have completed literature survey. Okay. So this is a paper. It is written here. Okay. So how many people you have seen? No, sir, this one. This one paper. Everything is here. Then I used to ask whether it is novel. Yes, sir, it is novel. Because it is not quoted here in this paper. He has only one paper. you record only one paper. And he says that whatever I am doing is knowledge. This is all about literary survey people are doing nowadays. Right? So this one should not do like this. So that's why the journal club is there to understand the research paper. How to implement this? How to implement this? You can select a research paper or a guide can allot student some research papers or individually or in a group of 2 or like that. Then that student has to read whole paper properly. Not just read, understand that whole paper. Right? Then make some notes on that. Whatever you understood from that paper. And then see or present whatever you are understanding in your own words. That is what is a journal club is. Then the rest of the people will counter him. Whatever his interpretations, whatever he want to say. If at all there is a misunderstanding or something else, or guide can tell him if whatever wrongly if he has stated anything. So this is what is to understand or how to read the paper and all this. If it is happening, very good. If it is not, see that it to happen so that it will be helpful in your overall development. Next. So the same thing I told that seminars in semester 1, semester 2, the seminars should be from your syllabus topic, and this is for the overall again a development of your confidence, presentation skill, and everything. So properly, if seminars are organized, you will see the transformation in the student. The first year seminar and the final seminar. If you compare of same student and if it is implemented properly with proper plan, the 100% transformation can be observed in the student. Right? So this is the purpose of seminar. Why some authority, some body, PCI or university or the experts, those who are designing the curriculum, why they are making all this? The reason is the overall development, not just to sell the degree certificate. Okay, next. Journal club. Same thing I told that how to implement it. Select paper. See how many of you know the chemistry people? Please raise your hand. Chemistry. Okay. Anyone tell me the name of any journal that you have seen of medicinal chemistry? Name of journal. Anybody? See what I expect? Dare to give a wrong answer. What I say? Dare to give wrong answer by standing up. Because someone failed 10,000 times, that is a wrong answer. He gave 10,000 times wrong answer. So he became a great scientist. Here at least, if you dare to stand up and say something wrong, it will give you a development. It will add to your development. That's why I say, tell me any journal that you know. Any journal, may not be of medicinal chemistry or anybody, not a chemistry people. Anybody, you just name a journal that you know, that you read, or any paper you read. Have you really done this? If not, then this means you are not in Pharm. You are still in a B Pharm and going to receive a Pharm degree. No doubt you will receive a degree, but you should be capable of justifying your degree. It should not happen that I am Pharm, I am PhD. हे सांगायची गरज नाही पडली पाहिजे. तुमच्या वागण्या बोलण्या दिसण्यातच जाणवलं पाहिजे तुमच्याकडे काहीतरी आहे. No need to show your file that I am this, right now. It should be expressed by your attitude. Attitude is not other sense, attitude. Getting? So, anybody knows? Journal's name माहिती आहे? चुकलं तर बोल.
International Journal of Pharmaceutical Chemistry and AI.
Yeah, okay. Who is publisher? Any other? मला आवडेल जर्नलची नावं ऐकायला. एखादं एल्सवेअर मधलं जर्नल सांगा. एल्सवेअर मधलं, स्प्रिंगरचं, विलीचं, बेंझम मधलं. हे कोण आहे? एल्सवेअर कोण आहे? स्प्रिंगर कोण आहे? कोण आहे? यूएसचा प्रेसिडेंट वगैरे आहे. कोण आहे? That is required. See that you which site search engine you use for literature survey? Where you used to do literature survey? The second year students, you must have done some literature survey. Some literature survey. काय वापरलं? Google Scholar. Okay. And शोधगंगा. Very good. काय शोधगंगा? इकडेच बोलता, मी इकडे येतो, तुम्हाला काय शोधून द्यावं? शोधगंगा काय? शोधगंगा गुगलवर काय मिळतं? किती पेपर बघितले? गुगलवर काहीतरी आपण कीवर्ड टाकल्यावर काय सर्च रिझल्ट किती पेजेस पर्यंत गेलं पुढे? समजा आपण सर्च केलं क्विनोलोन्स, अँटी हायपरटेन्सिव्ह, काहीतरी कीवर्ड टाक, किती पेजेस आहेत? किती आहेत? आपण सर्च केलं अरिजित सिंग, किती येतं? किती पेजेस आहेत? मग किती पेजेस बघतो आपण? 1, 2, 3 का 1, 2, 3 सर्च करतो रिझल्ट. नॉट मोर दॅन दॅट. दिस इज नॉट लिटरेचर. Right? So see that. So I am not. See, this is one hour or one day is not sufficient to tell and teach everything. Right? That's why I said you must read or you must listen in between what I want to tell. Right? And please implement. Next. Proposal presentation. It is there in the 3rd semester. Here. You have to present the outline of a research that you are going to perform. Right? So here, in consultation with your guide, you have to finalize. I am very much patient. Right? So you have to, in consultation with your guide, you have to finalize your research topic. You have to finalize your title. Then you have to show your line of action or plan of work. Get it approved and perform accordingly. That is what is purpose of the seminar which is there in the 3rd semester. Next. Now, this is the core part which you have to perform. Even though your course structure says that research to be performed in the 4th semester. But what I say is research is presented in the 4th semester. You have to perform for throughout 2 years. Then only it is possible to show some outcome. Otherwise here in the 4th semester only it is presented. Right? So whatever right from beginning, whatever title you have finalized, whatever your objectives you have set, accordingly you have to do some experimentation, data collection, finalization, and prepare your thesis, whole presentation, and submit finally. Okay, next. Finally, you submit your thesis, then it's examination, of course. So for that purpose, your examiner, external examiner is appointed. The guide is your internal examiner, like whatever it happens with the practicals. The examiner appointed outside, he is your or she is external examiner. You have to give your presentation in front of both examiners, and then evaluation is done. That will give you, that is called as viva voce, and your result comes out. So this is how, right from beginning, right from day 1 you admit, and this is the last point, that viva voce and your informed is over, you will get result like that. Next. In addition to this, there are some credits for co-curricular activities. Do you know this? If not, everyone should properly read first all curriculum. What is to be done, right? So this co-curricular activities means there are some weightages. You attend some seminars, conferences, workshops, national workshops, or national conferences, international conferences, or you present your research in this. So some weightage is there. Unless you do this, you may not get your result. So that is important component that has to be done properly. You suppose you have done some reviews or paper published like that. So in curriculum, the weightages are given for each activity. So accordingly, the weightage and marking is there that can be added, and that is there in your final result. So that is required. Next. Now, to monitor all this, there must be some program committee. The program committee, the main or chairman of the program committee must be some senior faculty. Then few faculty from each specialization, one faculty from each specialization. And in addition to that, the students should be involved in the program committee. Then this program committee will monitor whether the program is being conducted as per PCI regulations or not. So, to check everything, the program committee has to be constituted and it has to be monitored. Next. See, this is internal, external assessment. This is the common part that is known. So, we may not waste some time in this. Next. What not to do? We are towards the end of morning presentation. What not to do? The first, avoid plagiarism in assignments and research. What is plagiarism? Copying. So avoid this. How to check this plagiarism? There are some softwares that can check whether whatever you are presented is similar or not, plagiarized or not, copied somewhere from that, not. The copying may be from some existing material, or nowadays some artificial intelligence is there. Right? So now these softwares are also checking whether it is taken by or it is made by AI or what. The same thing, not only just copied from the existing content, but if it is AI assisted, AI supported, that also some softwares are there. Turnitin is there, right? Drillbit is there. These are the standard softwares that checks whether the material or similarity check is there, right? So even the AI based content, if it is taken, then it can be detected and it will not be allowed. So there is some less than 9%. Your similarity should be less than 9%. That is only accepted. So you have to see that it is done, right? How much AI you are using in your routine work, this college works, seminar, slide presentation, वगैरह AI वापरता, right? Those who are not using AI means they are not smart. They are fools. Don't give up their video. If AI is giving something in a minute, why to waste a day? Right? So it is good to know. Use of AI. Smart work is better or smart work is better? You have to use AI or not? You have to use, but what is but? You can have some support, but you just as it is, if you copy whatever supplied by that, it means you will not grow. You will be dependent on someone else. Your brain, think of your brain. If it is idle, anything idle, evolution. माकडांची शेपटी काय केली? माणसांमध्ये शेपटी आपण माकडांचे असो बहुतेक संबंध. मग आपल्यावर शेपटी नाही आली. त्यांची का त्याचा वापरच नाही. तसं मग काय होईल? आधी ब्रेन जाईल, ब्रेन नाही म्हणजे डोके जाईल. So think whether to use, how much to use. Smart work is good, but it should not make us dependent or weak. That you have to see that you should grow properly. So the plagiarism should not be there. Don't delay research planning or data. See, there are timelines. You have to submit your final thesis by this date. If you are delaying your experimentation, if you are delaying your data collection, everything, you may not be able to submit your thesis in given time. If it is not, then it may lead to the 6-month extension or like that. Right? So see that everything is in time. Avoid unethical research practices. Unethical research practice. Ethical? What is the meaning of ethical in Marathi? Exact meaning? तत्वा तात्विक बैठक असायला लागते. आपली तात्विक बैठक असायला लागते. मला वाटतं त्याच्यावरती जास्त अजून बोलायची गरज नाही. 2 वेळा म्हटलं आपली तात्विक बैठक असायला लागते, 3ऱ्या वेळा म्हटलं मी संपलंय. याच्यात सगळं चालू. हे सगळं वरचं खालचं काही गरज नाही. तुम्ही प्लॅगियरिझम करू नका, कॉपी करू नका, AI वापरू नका, हे सगळं सांगायची गरज नाही. एकच वाक्य, आपल्याला काहीतरी स्वतःचे प्रिन्सिपल्स तत्व असले ना की बाकी काहीच होणार नाही, सगळं व्यवस्थित होईल. राईट? ओके. डोंट रिलाय सोली ऑन गाईड्स. टेक इनिशिएटिव्ह. प्रोजेक्ट कोणाचा आहे? मग गाईड भेटले नाही, 2 वेळा गेलो, सुट्टीवर होते, मग आले तर मग ते बिझीच होते. असं असेल तर प्रोजेक्ट गाईडचं नाही. बरं, त्यामुळे डोंट रिलाय ऑन गाईड. आपलं काम करायचं. गाईड, व्हॉट इज रोल ऑफ गाईड? व्हॉट आय टोल्ड, गाईड he or she is there to just encourage you and just see that you are on the right track. Right? He is not or she is not there to work on your behalf. Right? So don't expect like आजकाल अशा अपेक्षा असतात तर माहिती नाही. म्हणजे कसं मुलांना अपेक्षा असतात, आई वडिलांनी सगळं करून द्यायचं. आमचं पुढचं आयुष्याचं सुद्धा आई वडिलांनी करून ठेवायचं, मग ते करणं. तसंच मग गाईडकडनं पण अशाच अपेक्षा असतील. आमचं प्रोजेक्ट पण आई वडील आमचं सगळं आयुष्याचं करतायत, तर मग गाईडने आमचं प्रोजेक्ट करायला काय पाहतो? आणि आई वडील पण करतायत ना, पुढच्या पिढ्या 7 पिढ्या बसून खातील असं काहीतरी करतात. तसं गाईड पण मग गाईडना पण असं वाटतं त्यांना की मग गाईडने पण का करू नये, आमचे 7 प्रोजेक्ट करा. आमच्या आई वडील 7 पिढ्यांचं बघतायत, तुम्ही कमीत कमी 5-7 प्रोजेक्ट बघताय. नो, डोंट एक्सपेक्ट लाईक दिस बिकॉज इट विल रुइन यू. हा तुमचाच प्रॉब्लेम, त्रास तुम्हाला होणार. ओके, सेम अवॉइड लास्ट मिनिट रश एक्झाम. ओके, दॅट्स इट. ओके, नेक्स्ट, व्हॉट टू डू. अटेंड क्लासेस, लॅब्स, seminars regularly. I hope. Okay. Looking at empty benches and all, waiting and all. I hope. Read syllabus document and course outcomes. जर केलं नसेल तर करून घे. Maintain research notebook. I have already told you that you maintain your notebook or register regular activities and get it verified from your guide. Communicate regularly with guide and faculty. That is most important. I always say that whatever you learn from your guide or mentor or faculty. So be in contact. Be in contact with them. Participate in general clubs, departmental activities. This is for your overall development. Overall development is required. Just getting some distinction or this marks, this GPA, it is not sufficient. If you go in the market मध्ये काय होतं का, मी इतक्या वर्षापासून गेले नाही. मग ते एकदम सांगतात सर तो त्याचं ॲडमिशन मॅनेजमेंट कोट्यातलं होतं, पण त्याला एवढं पॅकेज मिळाले 10 कोटी 10 लाख 12 लाख 13 लाख. आणि मी पण मेरिटने आलो होतो, मला ते एवढा स्कोर भारी स्कोर होता आणि मला अजून काय मिळत नाही. काय म्हणतात फर्स्ट इयरला कोणीतरी मॅनेजमेंट कोट्यात ॲडमिशन घेतलेला पोरगा आणि फर्स्ट इयरला कोणीतरी मेरिटनी टॉप मेरिटला आलेला पोरगा तो कंप्लेंट करतोय की सर मी मेरिटनी मला ॲडमिशन मिळालं होतं, पण मला अजून काय जॉब काय मिळेल झालंय. तो तर बघा तो मॅनेजमेंट कोट्यात आलं होतं त्याला चांगलं मिळालं. का त्याला असं म्हणतं मॅनेजमेंट कोट्यात आलं म्हणजे तो म्हणजे हुशार नाही आहे मुलगा, मेरिटचा नाही, पण त्यांनी कष्ट केले त्याने काहीतरी केलं असं स्वतःची डेव्हलपमेंट केली. नुसते मार्क म्हणजे काय बऱ्याच केसेसमध्ये जे मार्कशीटवरती डिस्टिंक्शन असलेले टॉपर असलेले टॉपरच्या यादीतले बरेचसे आणि बाहेर परफॉर्म करणारे हे सगळं वेगळं आहे. हे जर तुम्हाला माहिती असेल याचा अर्थ असं नाही की आपल्याला मार्क नको अभ्यास नाही करा असं नाही, ते पाहिजेच, ते पाहिजेच. त्याच्याबरोबर ओव्हरऑल डेव्हलपमेंट इज मस्ट. ओनली मार्क्स इज नॉट सफिशिएंट. Right? So you try to participate in everything, each and everything that will give your overall development. That is required. Stay updated with recent scientific literature, meet deadlines for submission and presentation. These are what things you should do regularly. Next. Thank you. Thank you very much. Now you can ask questions. मला असं वाटतं जेवढं हेवी सेशन काहीतरी सांगितलं होतं बहुतेक मला तेवढं हेवी करायला जमलं नाही. पुढच्या सेशनला या मध्ये एनईपीने आपल्याला दिलंच आहे ना, एनईपीने फ्लेक्सिबिलिटी दिलीच रिजनल लँग्वेजची असायला पण पाहिजे. का असायला पाहिजे माहिती का असायला पाहिजे म्हणजे आपलं पोचलं पाहिजे काय म्हणजे काय एकदा महाराजांच्या दरबारात बघा म्हणजे गुराखी असतो गुराखी त्याचा काहीतरी मोठा गुन्हा होतो आणि त्याला शिक्षेसाठी दरबारात बोलवतो. मग ते जे काही शिक्षा त्याचे सगळं ऐकून घेतात ते शिक्षा करते सर करा. मग तो शिक्षा सुरू करते त्याला काय शिक्षा सर करा. तो म्हणतो जे काय करायचं ते सरकार पटकन करा म्हणजे मला पुढे चालू जायचं. त्याच्यापर्यंत पोहोचलंच नाही, ती लँग्वेज तुमच्यापर्यंत पोहोचली नाही, रेसिपियंट पर्यंत पोहोचलं नाही, त्याला काय म्हणायचं तर त्या लँग्वेजला काही अर्थ नाही. पण पोहोचलं पण जर समजा आपल्याला इंग्लिश ही इंटरनॅशनल ग्लोबल बिझनेस लँग्वेज आहे ती यायलाच पाहिजे. अभिमान वगैरे सगळं ठीक आपल्या लँग्वेजचा मातृभाषेचा मराठीचा सगळा अभिमान असायलाच पाहिजे, त्याच्याबद्दल कोणाला शंकाच नाही. पण हेही मान्य करायला पाहिजे की इंग्लिश ही इंटरनॅशनल बिझनेस लँग्वेज आहे, ती यायलाच पाहिजे, नाही तर नुसतंच अभिमान बाळगत असेल. म्हणून खरंच आम्ही कोणी बसले असतील मी आमच्या इथे कोणी सेमिनारमध्ये कोणी मराठीत बोललं तर मी ऐकायचं नाही, त्यांना माहिती आहे काय लोकांना. मी म्हणत नाही आग्रह करायचं की नाही इंग्लिशमध्ये बोलत पाहिजे. पण ठीक आहे नंतर मला असं वाटलं की ठीक आहे तुम्ही बोला लँग्वेज तुम्हाला कम्युनिकेशन, पण याचा अर्थ असा नाही की आपण इंग्लिश सोडायला पाहिजे, इंग्लिश यायलाच पाहिजे नाही तर उपयोग नाही. ओके काय विचारायचं आहे किंवा मग आता प्लॅन करा काहीतरी विचारायचं पुढच्या सेशन पर्यंत. ह्या सेशनचे प्रश्न पुढच्या सेशन मध्ये विचारले तरी चालतील, वेळ आपण तयारी करायला थोडा विचारायचं नाही विचारायचं. यू कॅन हॅव सम टाइम, यू कॅन प्रिपेअर, विल मीट इन नेक्स्ट सेशन. ओके थँक्यू.
Take your lunch, come back, we'll exactly assemble here by 1:30, right?
जर तुम्हाला काही म्हणजे माझं पेस स्पीड इफ यू वांट टू गिव्ह सम फीडबॅक यू कॅन गिव्ह युअर टीचर सो दॅट आय कॅन इम्प्रूव्ह इन नेक्स्ट सेशन.
But next session is very important session where sir is going to discuss about general communication, thesis writing, results interpretation, everything. The main session is actually the next session. Please come.
preparing research proposal? Let us see. Next. Because we are a pharmacy students, we have to prepare some project, research proposal for pharmacy. How to start with, what to do? So first, we have to think about in which area I want to work. See, suppose you want to prepare a project for your, say, academic purpose, or suppose you want to prepare a project for any funding agency. In this presentation, I will cover the proposal for funding agency also and proposal for a student for their particular degree. So when we want to prepare any research proposal, we have to identify what are the first areas where I want to work. If you look at the areas given here, like cancer or diabetes, cardiovascular, anything, I hope, being a postgraduate student, you are very well aware about whatever areas or whatever diseases you have reported here, whatever severity of these diseases and how these diseases are affecting a whole community. I hope cancer, diabetes, cardiovascular, everything is known except microbial resistance. Or it is known. What is microbial resistance means what? Is it a thirst area? Is it a need presently? See, there is abuse of antibiotics. There is huge abuse of antibiotics or antimicrobials, I say. What is the meaning of abuse? Unnecessary use. Unnecessary use. See, everybody is suffering from any viral infection or throat. He used to or she used to go to medical and ask for AHIL. As common. No, everybody can get easily AHIL. And the situation now, or it is existing, maybe those who are directly working with patients and in the medical shop, they must be knowing that AHIL is not giving any effect. Right? People used to come again. I have taken AHIL for 5 days, no effect. Now then doctor gives some amoxicillin like that. What does it mean that now azithromycin means microorganisms are resistant to azithromycin. It is of no use nowadays. Earlier, when we were in schools, in that time, the brand which was working, that was Septran or Bactrim, which was containing sulfur drugs. Sulfamethasone and trimethoprim combination. Nowadays, we don't see these brands in the market. Right? Have you seen any time sulfamethasone and trimethoprim combination? No, it is not working. Right? So it is, the pathogens are resistant to this. Likewise, if this thing happens with this rate, if antibiotics are becoming ineffective in near time, no new antibiotic is coming in. No new, I am saying. Whatever coming in, analogs, penicillin analogs, cephalosporin analogs. New generation is not coming in. If situation is like this, means in near future there will not be any antibiotic available to treat a simple microbial infection. So people may die because of simple microbial infection because no treatment available. This is a major, I say the microbial resistance is a major issue nowadays. All others are major, definitely, no doubt about that. But this is one more thing. And alternative medicines. What is alternative medicine means what? Other than allopathic medicine. People are searching for it. If you are able to give best option or comfortable option or option with minimum side effects. See, major problem with modern medicines or allopathic medicines is their side effects, which are not known. Those who have taken the remdesivir as a Sanjeevani booty with paying high cost. How much? High cost. Right? in a single day, it was banned. Right? How come it is possible? Any Sanjeevani booty banned instantly, instant ban. Instantly it was observed that it is so toxic. Then what about those who have already taken? What happened to that? Many things are there, many issues are there. Let us not put leg on our right? So it's running back. But that's why morning I said ethics, ethical practice. Those who have some ethics, avoid this. Okay, forget about that. So if anything alternative can come out with better result, then people are searching for that. This can be one of the first areas for the research. Next. See, now when we think about development of drug, how drug can be developed? Where from drug comes? What are the sources of drug? See, drug can come from natural source, plants, microbes, animals, or it may be by some chemical synthesis. Agree? So what is drug? What do you mean by drug? When I say drug, means what drug? Morphine, codeine, heroin? You are talking about that drug? Or what is drug? Any chemical is drug? Benzene, nitrocyanol, toluene, is it drug? Then what is drug? Benzoic acid is drug? So drug is something that is found safe and efficacious and approved by regulatory authority. Then it becomes drug. Not everything is drug. So what is the source of drug? Drug may come from plants, or from microbes, or from animals, or by chemical synthesis. It may be any chemical entity. But a chemical entity, every chemical entity cannot be a drug. So what happens when you want to get something from any source, say from plant source? Suppose you want to identify a drug, or a drug-like entity. You identify drug-like entity from a plant source. In that case, what you have to do? You have to identify some plant, right? Then extract the chemical constituents from the plant, right? Then after extraction, isolate that chemical constituent. How many constituents contain any plant? Do you know? A plant can contain how many constituents? N number. Don't. Multiple number of chemical entities may be there from the plant source. Okay. So after extraction, isolate individual chemical entities. Then identify that individual chemical entities, that is characterization, structural interpretation. Right? Then isolate in pure form. Then test on the animals, that is called preclinical trials. Right? If it is found safe and efficacious in animal trials, then go for clinical trials. Phase 1, phase 2, phase 3. If it approves, then FDA approves that it has a drug, enters into the market. Right? And then phase 4 trials, post-market. This is how drugs comes in market. I have given example for a chemical entity coming out of plant. The same thing for, say, coming out of microbes, coming out of animals, or coming out of chemical synthesis. Microbes are giving drugs. Sure? Can we get drugs from a microbe? How? Do you know any drug which has came from the microorganism? Is there any drug that microorganism gives? Huh? You are brilliant boys, right? Yes, all antibiotics are coming from microorganisms. Microorganisms gives drug. Sure. Likewise, any source can give chemical synthesis. Yes, most of the compounds which are synthesized chemically, then tested. So you got the all route how drug is developed. It can be from any source. Identify that. Test. If it is safe, efficacious, get approved, it becomes a drug. Right? Once it is approved, then you can formulate it in suitable formulation. Paracetamol. Paracetamol is a drug? Yes. Now, we take powder paracetamol? Then, here the formulation development comes. Right? API, active pharmaceutical ingredient identification development, it is part up to this. Okay, paracetamol identified as a safe, efficacious as an antipyretic, approved as an antipyretic drug. Okay. Next step comes administration of that. So, what are the formulations available for paracetamol? Only tablet? Syrup? Only syrup? So, as per requirement of patient, if a kid, small kid, he can't have solid dosage form, so liquid syrup is available. Okay, suppose a normal person can have a solid tablet, he can swallow. Okay, if he can't swallow anything, bed injected, right? As per requirement. So this is what is called as formulation development. Those who are in physics, they can think of this. Does it mean that only physics can think of formulation development? No. Anybody can do anything. It is not like if this specialization will do this like this. No. If you have that skill, knowledge, everything, anybody can do anything. Agree? Okay. So identification of drug, molecule, active pharmaceutical ingredient, and formulate it into suitable formulation. Here R&D is divided, like say basic research and formulation development. You must have heard about R&D, basic research and development, F&D, formulation development. So the difference is, a basic research means drug discovery. F&D means formulating that identified or developed drug into suitable formulation. Then one more thing, analytical research. What is analytical research? Right, analytical method development. It is also there. Whenever new things are coming, new drugs are coming, new impurities are identified, new degradants are coming in. So that for that purpose, new analytical methods development is required. So this is one more area. Have you heard about Rantac? Ranitidine? What for it is used? And is it there in the market now? Some few days before, you must have heard the news. It is banned. It is banned because some carcinogen impurities are identified in that. So it is continuous development. Because ranitidine have some impurity that is carcinogenic, that's why it has been banned. So who identified that? How it was identified? What is the method for identification? Well, these are analytical methods coming in. Okay. Now, the research basically starting with first identifying drug by any source. So we can say the phyto research, phytoconstituents, chemical constituents from any natural source, synthetic chemistry, anything. Once it is identified, it is prepared, its characterization, structure elucidation, where all analytical methods comes in. Once it is identified, then its testing, animal testing, pharmacology research comes in. Preclinical trials, studies on animal. So how studies on animal is carried out? Do you know anything about how animal studies are designed? See, in this session actually we will have some rough idea about everything. So when we think of animal experimentation, suppose you have synthesized new compound, any new compound, and you want to test it for its any activity, say anti-diabetic activity. So how to design this, how to perform this, what is required? For that purpose, very first thing that whatever you have synthesized, that compound is required. Second thing, animal, you have to decide some model on which animal you want to test. Initially, you have to start with small animals, either mice or rat. So, the smallest animal used for testing is mice. The very first thing you have to do is, you have to first find out its toxicity. That is its LD50. Lethal dose. Minimum lethal dose, right? So how to find out lethal dose? LD50 means 50% of population when it dies. So that dose we have to calculate. Means what, suppose you have to take 6 animals, 6 mice, you give a dose in rising, say 1 mg to whatever, right? And identify a dose at which 50% population dies. It means its LD50 value. This is how you have to find out its LD50 value. Then you have to find out its doses. Right? So you have to start with particular doses and identify whether it gives effect or not. Now when we are testing for anti-diabetic activity, the very first thing you have to do is, you have to induce diabetes to the mice. Anti-diabetic activity cannot be observed in the normal mice or normal rat. So for that purpose, we have to induce diabetes. So some diabetes inducer is required. What is that? It may be streptozotocin, it may be alloxin, whatever, right? So you have to induce first diabetes, make them diabetic, then treat with your compound, and then check whether it is showing anti-diabetic effect or not. Is it okay? No. Still you have to need something for comparison. Some standard compound is required. So known anti-diabetic, say metformin. So you have to compare the activity of compound which you have synthesized with a standard drug which is known that it may be metformin. It means you need groups of animals, standard control, right? Then only vehicle control, then your test group. So how many animals required in one group? Statistically, to be a statistically valid? 6, right? So I think you are known with this, right? So one group of 6 animals is required, minimum. Why 6? Why not 1? To average out, validation, right? 1, it may give false result. That's why statistically valid result. Suppose you want to have a statistically valid result, you need a multiple number than single. When you go for titration itself, you used to take 3 readings and then have mean. Right? That's the reason. A single reading may be erroneous. That's why multiple reading, and to be a statistical valid, we need a multiple readings. That's why we need a multiple animals. Got it now? So by any means, when you identify new molecule, you have to isolate it, structurally characterize it, then go for its activity testing, animal testing. Now, once it is found effective and safe in animal trials, then it goes to the clinical trials. Animal trials, it is called as preclinical trial. Clinical trials means trial on human. Now, when you are going for trials on animal, it needs permission of some committee. You know this? Animal Ethics Committee. Institutional Animal Ethics Committee. So there should be a constitution, proper constitution of Institutional Animal Ethics Committee. There should be proper animal house. Then you can perform this. Otherwise, you can't perform like anything, get with some animals and test. It is not allowed. It is a crime. It is a crime. So it is not allowed. Now, once in clinical, preclinical studies or in animal trials, when it is found safe and efficacious, you go for clinical trial in humans. Now, very interesting. Suppose you have to give a dose to first human being. How much you will give? Are you getting what I mean to say? A new compound. You have tested that compound in animals. Okay, you found it safe. You found its LD50 something X. It is efficacious. Okay, it's a rat or mice. Okay, cat. Suppose you want to give human. And together. Imagine. So how to start with? Is it safe? एवढी गंमत आहे. कोणाला तरी एखाद्या जिवंत माणसाला काहीतरी द्यायचं आहे. ते माहित नाही आपण काय कसं होईल त्याच्यावर कसं काय होईल. That is those okay, but you have to start with 10%, 1/10 of LD15 animal. What I told? 1/10 of LD15 animal. That much initial dosing we have to start with when you have to give the human. So safe, very safe. Then gradually one can increase. Do you know how much time it takes any drug to develop? It may take 10, 15, 20 years. That is the reason. But you know, the vaccine were developed in years. Surprising. Better not to comment on that, whether it was emergency or what. Okay, you see, now it's emergency to those who have received it. That is the main issue. Okay, so I hope you are clear with this whole process. New entity, its testing, efficacy, clinical, preclinical, clinical. Then once it is approved, then you can think of its formulation. Okay, now you can prepare proper formulation, suppose tablet, capsule, whatever. Then that formulation needs to be tested also. Okay, before that tell me, suppose you have given powder and you have formulated. Suppose how you will start? Formulation development. How will you start? Suppose you have to prepare a tablet. Forget about what you have to prepare. Suppose it has been decided that I want to make a tablet of X material. How to start? What is the first thing that is to be done? What is it? It is called the pre-formulation. Pre-formulation studies. Simple. Means what? To form a tablet, you need API. Say it's a paracetamol, you need paracetamol. Then what you need? Whatever additives. So binder, then additive, whatever. So whatever you need, A, B, C, D things, you have to first check its interaction, whether they are compatible to each other. That is called the pre-formulation stage. Once they are compatible to each other, then you can formulate, you can make A, B, C, D, A to Z formulas, different combinations, right? Then you will check its evaluability for all the evaluation parameters. What are evaluation parameters for tablet? Okay, everything, hardness, disintegration, dissolution, whatever. So you will evaluate for all the formula which is suitable, which is found perfect, then you will select and you will formulate it. Right? Once it is formulated, then again test it. Just testing by dissolution or disintegration, these are called the in vitro studies, out of body. It will not suffice. Right? Then you have to administer it to the say first animals, then get whether it is safe or not. If it is safe, then go for clinical trials again. Clinical trials for that formula. Then if it is safe, efficacious, everything, then it will be approved as a that drug. It can come in the market. Then given to the patient and all. This is whole about a summary of, I can say, a drug development process. How a drug from research to drug to the patient. How it goes. Now here, various research components are involved. Right from basic research, analytical development, then pharmacological investigation, clinical investigation, post-marketing investigations, market research, everything needs, what is research? which is existing already. Repeat search. It is existing. We have to again find it. That's why market research. Now people say, what is market research? Market is already existing. Then what you are doing? Analyze all these situations. Collect the data. Again, in the morning I have shown the experimentation, data collection, data analysis, then interpretation, then your conclusion. Right? This whole process is a research. Anything. So it needs every pre-experimentation, experimentation, post-experimentation, data generation, data collection, data maintenance, data analytics, and conclusion. So this is the whole process that you have to do. Okay? Next. Now, just for information, if this is one example we have taken, the drug development from the plant source. So what is required for that? You need to select the plant. Why I am talking about plant, you know? Next please, one slide. Look, whatever drugs available in market, 40% of the drugs are derived from the plant source. Again, quinine, all anti-malarial drugs, artemisinin, aspirin, many, you take a name, it is coming from the plant source. Are you getting? Though they are chemical entities, they are chemical entities, simple single chemical entities isolated from the plant. Right? That's why I am talking about this. This is the area actually that needs to be explored. Chemical synthesis is okay, though I am from medicinal chemistry. I like it very much. I like synthesis very much. But I also know the fact that how much outcome is coming out merely by only synthesis. There is very limited outcome coming out. So this is a natural source. A ready drug is coming. Artemisinin, you have heard of artemisinin. Artemisinin. What for it is used? It's anti-malarial. Recently, some Chinese scientist discovered it. She got a Nobel Prize for that. Right? So it is from the natural source. Likewise. So new drugs are coming like this instead of synthetic route. Or you can go for semi-synthetic route like modified natural analogs. Are you getting? Means what? Natural analogs, you structurally modify that. Simple one-step, two-step modification. That can be a new molecule coming out. Right? Natural molecule is formed, safe. Just go for its computational studies, identify what properties to be changed, accordingly put some substitute in that, and that could be a new entity. Likewise. So, if you want to go for drug discovery from the plant source, you need selection of plant, then authentication of plant. What is authentication means? Selection of plant, okay. And then authentication means what is this? Plant is plant. Tulsi, okay, Tulsi. Everybody can know this is Tulsi. So she may say this is Tulsi, he may say this is Tulsi. Then what is authentication? Or is there any difference between this Tulsi and this Tulsi? Black Tulsi, white Tulsi, this. There are various varieties, species, genus, different species in the same family. It may be the same family, but various varieties. Right? So identify if the species changes, then all chemical constituents are changed. So who will authenticate it? The person who is botanist. Right? He can only identify what it is. Suppose you have given a paracetamol, what you will do? A powder, and identify it. You will go for spectral characterization, check what is melting point, what is IR, NMR mass. Then you can say no, it's a paracetamol. Okay. Likewise, suppose you say it's a Tulsi, then someone should authenticate it by having all tests and check, and then he will label it. No, this is this. Right? Then this is what is authentication. Again, it needs a competent person. Authentication from the competent person, not just someone says and you will. Then extraction and isolation. Extraction. You know extraction, oxalated, solvent extraction, maceration, cold extraction, there are different methods. So from that plant source, again, when you are doing from the plant, there are various components of the plant. Root, stem, bark, leaves, whatever. So by literature or by records you can know which is the component of that plant that is containing the required chemical constituent. You can use that particular plant or even you can use whole plant, whatever. As per the depending upon which plant you are using. Go for extraction. Extraction, various solvents as per polarity and all that. Then separation, fractionation, identify different constituents. Then structural elucidation, characterization. Then biological screening, whatever I told, like it's like a new chemical entity, screen for its activity, which activity it shows, and accordingly, if you want, you can go for molecular modeling studies, like talking and computational data, whether by which mechanism it is showing a particular activity, that can be seen. You can go for synthesis. A similar molecule can be synthesized, or some structural modifications can be done, semi-synthetic analog. Likewise, penicillin. Penicillin is natural penicillin, benzyl penicillin. But amoxicillin is analog of penicillin. It is called a semi-synthetic analog of penicillin. Only one functional group is added, and it becomes the same or even better than natural penicillin. Likewise, any entity from the natural source can be structurally modified, right? And that is by synthesis, and that can be tested for. So this is how the path for drug discovery from the plant source. Same can be followed for animal source or microbial source, or a synthetic is a different issue. Next slide. So see, the plant sources, these many drugs, even what I said is 40% of drugs on the counter, they are of plant origin. Okay, next. Now, okay. Many times I have heard the word drug. Okay. So what is that? Something safe and efficacious. Important. It should be safe, it should be efficacious. No, what is the definition of safety? Do you know cancer treatment? The fellow who undergoes chemotherapy, the major side effect associated with cancer treatment is very severe. It loses hair, then weakness, every system goes like damaged like that. Suppose such a severe side effects, if they are associated with paracetamol, okay, we will take it, paracetamol. Why not? Because it is reducing your fever, efficacious. But so serious side effects. With paracetamol, suppose you are losing your hair. Okay? See, means the safety and efficacy balance is required. The cancer, end is death. So to avoid death, we can lose hair. It's okay, na? It's better to alive than losing that hair. That's acceptable. So such a toxicity, such a side effect, which are acceptable, that depends upon what efficacy, what purpose it is used. That's why anti-cancers with severe side effects can be acceptable. But a paracetamol like drug or a simple treatment, so side effects should be acceptable. The severe side effects cannot be likewise. Okay. So here we are talking about a drug action. How drug shows action? The drug is now formulated, a tablet. Say imagine a diclofenac tablet. We are visualizing effect of diclofenac tablet. Imagine, suppose you have a knee pain. You have a diclofenac tablet. Okay. Tablet, solved. Finished. Pain is over. Okay. Does it happen like this? See, imagine you have pain, you have tablet with water, solvent. It relieves your pain. है ना? होतं की नाही? असं होतं की नाही? गोळी खाल्ल्या थोड्या वेळ तरी पेन जातं की नाही? थोड्या वेळ जातं. लगेच नाही थोड्या वेळ. What is this? How it happens? What processes takes place after swallowing a tablet? Being a pharmacy fellow, we should be very clear with this. Otherwise, people may feel it's a magic. Have a tablet. एक गोळी खाल्ली, जिभेवर ठेवली, एक ग्लास पाणी पिलं, आणि अर्धा एक तासाने पेन गायब झालं. इथपर्यंतची प्रोसेस कोण सांगेल मला? काय होतं ते? हे विद्यापीठात येत तसं काय होतं काय होईल? चल मी परत एकदा सांगतो सगळं इथून पुढे लक्षात घ्या. सी टॅबलेट इज अ सॉलिड डोसेज. हाऊ टॅबलेट इज प्रिपेअर्ड? By first maybe by granulation or may not be by granulation, whatever. If required, you may prepare granules. If not required, you may not prepare granules. Now, when granulation is required, when it is not required, anybody can tell. What is required? What is safe compressibility? Kadi asa safe compressibility? So flow properties matter, right? Suppose you are given a sodium chloride powder. Suppose you are asked to prepare a tablet of sodium chloride. Which method you will use? What are different methods for tablet preparation? Granulation or direct compression. Granulation means wet granulation, direct granulation. Now, when you are given a sodium chloride, whether you will go for direct compression or granulation? Direct compression, simple. It has good flow properties, no need to prepare granules. Okay, so whatever. Now you are clear with tablet preparation. Now tablet is prepared. The patient is receiving tablet. Okay. First, when it's swallowed, where it goes? Stomach. What happens in the stomach? Release drug. Now here drug is diclofenac. In the stomach it will release diclofenac. What will happen? That diclofenac will there. It is there in the stomach now. It will be absorbed. Where? Blood. No. It is small intestine. From stomach it will be absorbed into the systemic circulation and in the blood. This is called as drug absorption. Suppose there is other drug. Forget now acetylphenone, diclofenac. Now there is something else that you expect it should reach to the intestine. As it is, what do you do? How? Sustainable is different. Suppose you want to protect which protein? Enteric protein. Those drugs which are acid sensitive. Acid sensitive means what? If they come in contact with acid, they will degrade. So that kind of drugs, if suppose you want to prepare oral tablets for that, then it needs to be protected from the acid environment. So it is coated with the enteric coating. What is the enteric coating? It will not break in the acidic coating. It will be intact. After 2 hours, normally transit time is 2 hours. After 2 hours, it enters into the intestine. In intestine, what is pH? Okay, basic pH. So, in intestine, because of basic pH, enteric coat breaks down. Then granules are released. Then drug is released. Then it is absorbed. Okay now? Okay. Now drug is absorbed in the blood. Now what happens? Drug distribution. Blood carries that drug wherever it goes. Agree? Where drug goes? Everywhere. Agree? So drug is distributed throughout the body. Get it? Throughout the body. But brain is protected. There is blood-brain barrier. It is a highly lipoidal membrane. So it will not allow many drugs to enter into the brain because of it is highly lipophilic. Only highly lipophilic drugs can enter. Agree? This is what is drug distribution. Drug is distributed throughout the body. What for you have taken diclofenac? Diclofenac कशाला घेतली? Drug तो चालला. काय गरज नाही. Getting? So, though you want it here, the drug is distributed throughout the body. Throughout the body. Okay, so as like it is also received to the knee where there is pain. Why pain? There may be some damage of the tissue. So what are the markers for pain? Tissue damage, release of arachidonic acid pathway. What is that sign of prostaglandins and prostacyclins? So arachidonic acid converted to prostaglandins, prostacyclins by enzyme COX. So how diclofenac act? How it gives pain relief action analgesic effect? Because it blocks enzyme COX. Cyclops. Okay. So what is COX enzyme? What is enzyme? Enzyme क्या है? Because what is enzyme? Is it acid, base, lipid, protein, carbohydrate, fat? क्या है? What is chemically enzyme क्या? These are proteins, chain of amino acids, right? So the COX is a protein, means chain of amino acids. Okay. See, right? Now the COX is there, which is converting arachidonic acid to the prostaglandins, prostacyclins. That's why the pain is. That is a symptom of that. Now what that diclofenac does, it goes to the COX and it binds to the COX and stop action of COX. Is it so easy? The COX is waiting. Come diclofenac, come bind. Find with me. Right? So when is the protein? What is the protein? Chain of amino acids. How many amino acids? And there will be 400, 300, 500 amino acids. N number. So it is a chain, like a thread or hair you can see. So imagine a hair in the water. A single hair, a long hair in the water. It is continuously changing its shape. Can you imagine? Hair in the water, dynamic. So likewise, your COX is in a dynamic state of conformation change. Now diclofenac, a small molecule, going to that COX. Now COX, because it contains 400 amino acids approximately, it may not be 4 exactly, then out of 400 amino acids, the diclofenac binds only with 4, 5 or 10 amino acids of that. Right? So where are these 10 amino acids, where diclofenac has to bind? Okay? Lock है, lock. अख्खं लॉक आहे आपलं उघडायचं आहे ती चावी फक्त त्या कीवर मध्ये जाते पण ते लॉक पण डायनामिक आहे एका जागेवर स्थिर नाही पण नॉर्मली तिथे लॉक स्थिर आहे आपण चावी घेतो लगेच उघडतो पण लॉक पण हलतंय कंटिन्युअसली चावी पण हलते कंटिन्युअसली मूव्हिंग ती कधी मॅच होणार कधी ते फिट होणार आणि कधी आणि गेटिंग दिस कॅन यू इमॅजिन सो दिस इज व्हॉट इज हॅपनिंग सो दॅट COX is also moving, changing its shape. Your drug molecule is also changing its shape. When both are complementary to each other, they will bind. Then it will stop action of that COX. Then conversion of arachidonic acid to prostaglandin cycles can be stopped, and pain will be relieved. That's why लगेच गोळी खाल्ल्या थांबत नाही. एवढा वेळ जातो अर्धा तास पाऊण तास या सगळ्या प्रोसेस आहेत. Got it now? So this is how drug shows its action. Okay, but is it possible for anybody to visualize what is happening in the body? No. That's why this plasma concentration curve is studied. Because we can correlate this with concentration of drug in the blood. Oh, okay. So here, this is the zero time when it is swallowed. So gradually its concentration in the blood increases. When it reaches to minimum effective concentration, it starts its action. Its concentration goes on increasing. Similarly, because of its metabolism and excretion, simultaneously when drug is absorbed, simultaneously it is metabolized also, it is excreted also. So at a particular time, its concentration will start declining. Right? So its concentration will decline. After some period, after say 6 hours or 7 hours, you may feel like pain again. So again you take another tablet. Are you getting? So this is the dosing interval. The dosing depends upon the duration of action. How long it shows action? Then after you have to maintain its concentration above minimum effective concentration. That's why the dosing interval is given. And higher dose, minimum toxic concentration. Means what? If instead of having one tablet, 100 mg, suppose you take 200 mg, what will happen? Its concentration will be higher. As its concentration is more, it may start showing some side effects, toxicity. If it is still higher, then higher toxicity, like that. So the concentration should be maintained in its desired therapeutic range. So dosing interval is decided based on this plasma concentration. Okay? Got it now? I hope right from drug to its formulation, what are the minimum steps to be considered while doing research? I hope that I have just focused, that I have summarized. Okay, next. So, drug discovery, methods of drug discovery. How drugs are discovered? Various examples, various methods are given. To discover drug, one needs first hit, lead, and then drug. Hit to lead, lead to drug. This is a way of drug discovery. So, these are the various methods for lead discoveries, lead identification. Serendipity, random screening, like this. So, serendipity, what is serendipity? Means it's accidental discovery of drug. There are many drugs, those who were discovered accidentally, without any plan. One example, very known example is penicillin, that Alexander Fleming. He got Nobel Prize for that. Who was Alexander Fleming? Medicinal chemist, doctor. He was a microbiologist. See, the microbiologist can develop a brilliant antibiotic like penicillin. Still it is working. So the qualification doesn't matter. When you say that I am from physics or I am from chemistry or I am from pharmacology, doesn't matter. You need a skill. See, he was a microbiologist. Look, penicillin still it is required. How many lives he saved during that World War? Before that, no antibiotic was there in existence. It was the first antibiotic, which was discovered accidentally. That's why I say curiosity and patience is required. How it was discovered? You know the story of this? Anybody know the story? How many of you know the story? You know the story. Rest you don't know. So I must tell. How I know? See, he was a microbiologist. He was working on Staphylococcus aureus. What is Staphylococcus aureus? Gram-positive bacteria. So while working, you must be knowing the petri dish, petri plates, then culture growth and like culture media. So once he decided to go on vacation for a week, he closed his lab and went for vacation. By keeping everything as it is, closed down shutter, went. A week after he came, he found that on one petri dish where he tried to grow that Staphylococcus aureus, on one plate there is some area where there is no growth of microorganisms. On one plate, particular area has no growth. He surprised. Upon getting he surprised, why not? Everywhere there is growth of Staphylococcus aureus, but there is particular area where there is no growth. Okay, he tried to identify reason. Then he found that the growth, where the area where there is no growth, there is some contaminant. Some foreign material is there, and around that, the microorganism of Staphylococcus aureus has not grown. Then he tried to find what is this. Why? What is this thing where around that there is no growth? Then he tried to find, and it was a fungus. Now again surprise. Fungus is a microorganism. One microorganism is not allowing to grow another microorganism, though he is not human being. Normally human being is open. One fungus, one microorganism is not allowing to grow another microorganism. How it is possible? Then he tried to find that fungus, what that fungus is. It was Penicillium notata. And why it is not allowing to grow microorganisms? Because the fungus, when it is growing, it gives secondary metabolites. These secondary metabolites are again chemicals. That is benzyl penicillin. Because of that secondary metabolite from the fungus, benzyl penicillin, there is no growth around that. So penicillin was discovered. Then he identified what is that secondary metabolite. Then he went for structural isolation and all that. Then it was benzyl penicillin. So this is how benzyl penicillin has been identified as an antibiotic. Now the research was not stopped here only. The benzyl penicillin, that is natural penicillin or penicillin G, it is only active by injectable. It is not stable in acidic environment. It is not stable in alkaline environment. So it cannot be given by oral route. So what happened? The scientists decided to work on this and prepare some acid stable penicillins, some alkali stable penicillins, so ampicillin, amoxicillin, proxicillin, diproxicillin, various penicillin analogs, those who are there in the market. These are the semi-synthetic penicillin analogs. Got it? So this is what is curiosity works. Okay. So likewise, even that diazepam is anticonvulsant. It is again accidental discovery. In the Roche, someone was working on the project. I will tell you this in short. He was working on the project. He synthesized 242 compounds and tested for activity. He found that no compound is active. So he terminated the project. 2 years later, during cleaning of laboratory, he found one vial which was not tested. So to complete formality, he submitted that vial for test and it was active. Then it was modified as a diazepam. This is again accidental discovery. Then random screening, non-random screening, metabolite studies, clinical observations. These are the various methods. Random and non-random is what? Random. Every chemical tested for each activity. If found active, okay. If not, forget. Likewise. So each and everything to be tested. It is of not use. It is very costly. Non-random is what? Suppose you have 100 compounds. Then group them on basis of their structural similarity. Right? Then suppose there are 10 compounds with same structure. Instead of testing 10, test 1. If it is active, test rest of 9. If it is not, forget. Likewise. So this is non-random. Metabolite studies. See, metabolism is what? Drug metabolism is what? Conversion of drug into its more polar so that it can be easily eliminated from the body. The metabolism process in the body is to facilitate its elimination. See, body doesn't accept anything foreign material. Anything comes foreign, it will try to eliminate it out by any mechanism. The one part is metabolism. Now you must have heard that fexofenadine. What does it? Brand Allegra, right? It contains fexofenadine. It is non-sedative antihistamine. This is actually metabolite of terfenadine. Look at this. Earlier, the terfenadine was used. It is non-sedative antihistamine as anti-allergic. Then it was observed that its metabolite, fexofenadine, is more active than terfenadine. So now in market, terfenadine is not there. Fexofenadine is there. Right? This is how the terfenadine is the clinical observation. See, Viagra was discovered by clinical observation. It was up to clinical phase 2. It was under test as a Anti-anginal drug. What is anti-anginal? Angina pectoris? Chest pain because of constriction of coronary arteries. Constriction of arteries. So what is required? Vasodilator is required. So beta blocker is different, but he phosphodiesterase inhibitor. This Viagra is phosphodiesterase inhibitor, right? So it gives vasodilatation. So it was under investigation for angina pectoris till phase 2. In phase 2 clinical trial, it was observed that it is effective in sex organ and not on the heart. So it is the research has been modified to its as anti-impotency, right? So this is how the clinical observation can give. Now importantly, rational drug design. Rational drug design. Earlier, whatever we have seen, either it was accident or random thing. Rational means what? You know target, you know ligand, you know lock, you know key, you can develop a duplicate key, something else like that. So everything is clear. The mechanism is known. Right? So rational drug design used. Where here the computers are in use. Right? It may be structure-based drug design, it may be ligand-based drug design, like that. So various drug design processes can be done. Detail
Okay. Detail.
This particularly, it is going for drug design process, right? Means what? Suppose you know the target, likewise diclofenac. Diclofenac is ligand and COX is a target. Now the COX, suppose you know you want to develop a drug which can inhibit COX. Now target is identified, it's a COX. Then means lock you know. Now suppose you have a lock but you lost a key. What do? You can prepare duplicate similarly. Right? Similarly, you can prepare different ligands and you can use computer softwares. You can test virtually. Suppose you have 1 lakh keys, virtual keys, and try which one is fitting. Instead of synthesizing, actually, virtually you can screen and then synthesize which is best. It will save your time, money, everything. This is what is the glory of this rational drug design. Or secondly, that is ligand-based drug design. Means you don't have lock, only key. You go to the key maker and then ask him to prepare duplicate. Likewise, QSAR, format to formatting. These things are like ligand-based drug design. Ligand is known. You have to develop a new drug, new ligand. This is how the structure-based drug design or ligand-based drug design can be done in this CAD or rational drug design. Okay, next. Okay, so this development process, if you look at, it starts here from target identification, function study, drug design and optimization. Target identification means what? Suppose you want to develop a drug for cancer. Okay, you must say earlier in the first slide, I talked about some thirst area, where suppose someone wants to work in the cancer. So what is first thing you have to do is, first study the cancer pathophysiology. What is cancer? Uncontrolled, unwanted growth of cells. Means cells are multiplying. Because of cell multiplication, there is a tumor formation also. In that case, you have to identify a particular step that if that step is stopped, the cell multiplication will stop. So that mitosis and all things, cell division, you know. Right? So in that case, suppose you identify a particular target, like in inflammation, what I told COX, likewise in cell multiplication, cell division, there are multiple enzymes which are taking part. It may be DHFR, it may be topoisomerase, it may be polo-like kinase, it may be EGFR, many targets, n number of targets are there. So what you have to do is, you have to identify which target you want to inhibit, which target you want to act on. That is your lock. Identify that target. Then start designing your keys, your molecules, or it is called ligands, that can fit into that target. If they get fit into target, it will stop that target to do its normal function. So, the cell multiplication, cell division will be stopped. So, no unordered growth of cells and everything. Clear? So, this is how the target identification, functional study, drug design and optimization is done. Got it? Okay. Then once lead is identified, then it needs to be optimized. Lead, what is lead? Lead means a compound, it has required biological activity, but it is not safe. It may not be safe. It may have some toxic effects. So, the structure of that lead needs to be optimized so that it will show its required effect, but it will show minimum toxicity. Okay, leave it. See, suppose this is the key, right? If this is the key, when I want to open the lock, which part of the key is required to open the lock? Only this part. This part only enters in the keyhole, it opens. So this is called a pharmacophore in case of drugs. Right? Only this part is required to show the activity. Then what the hell all this is? See another key. Only this part is required. What the hell rest of the things are required? Yeah. Why to increase the cost? Getting? प्रत्येक जर एवढाच पार्ट आपल्याला गरजेचा आहे, स्ट्रक्चर मध्ये एवढे 2-4 फंक्शन लूप गरजेचे आहेत, तर आम्ही 10-20 कशाला काय करतो? आहे का नाही सांग का exactly. Suppose you only prepare this part, how to hold it, how to bring it to this side of action, right? This means the pharmacophore of a drug, it is a part of the structure that actually interacts with the target, but the rest of the part is required to bring it to the target. Here, नसेल तर आपण लॉक पण घेऊ शकणार नाही. So bring it to the target. So rest of the part, and because of this thing which is there in addition, it may give some toxicity. So lead optimization is required. Getting it? This is how lead optimization is done. So functional genome, drug target validation, lead discovery, lead optimization. Once lead is optimized, go for preclinical. Preclinical means if it is okay with animal trials, go for clinical. If it is okay with clinical, it will pass. It will be approved as a drug. It will enter into the market. That's all. It's simple. Because life will be drug. But imagine time, cost, manpower. तुम्हाला माहिती आहे एवढ्या दिवसात तुला नवीन ड्रग आलाय तरी जरा हे झालं लेक्चर आपण एक एक्सरसाइज करा मागचं सगळं लिटरेचर वाचा आणि लेटेस्ट नवीन ड्रग कधी आलाय कोणाचा आला नवीन ड्रग नवीन फॉर्म्युलेशन किंवा एखादा अॅनोलॉग पण नाही जसं ॲमॉक्स आला त्याला काही म्हणता येणार ना कारण ते पेन्सिलिन अॅनोलॉग त्यामुळे त्याला आपल्याला नवीन मॉलिक्युल नाही म्हणतो एम्पिसिलिन ॲमॉक्सिलिन हा पेन्सिलिनचा अॅनोलॉग तो नवीन नाही असं नवीन काहीतरी काय आलंय का जरा काळा माहिती मॅडम सांगा मॅडम मला सांगा मुलांनी ह्या शोध लावलाय हे नवीन काहीतरी आलंय आता माहिती घेतली ओके आपण बघूया काय येतं कधी येतं कसं येतं आणि मग आपल्याला काय पडायचं आपल्याला काय काम ओके चलो नेक्स्ट ओ चल इज इट विजिबल ओके लेट इट सी
वाइट कर
to stop the action of PLK1. The cancer multiplication will be stopped at 3 stages. It will be more effective. Inhibiting one step, it is better to inhibit at 2 or 3 than 1. Of course, synergistic effect. That's why we have identified the PLK1 as our target previously. Okay. So what is this PLK1? The PLK is a family. Out of that, 1, 2, 3, 4, 5 different PLKs are there. Out of that, we have selected PLK1. Again, like plants, we are saying this is tulsi, this is tulsi, this is tulsi. Very different varieties. So you have to select particular. So PLK is a family. Out of that, you have to identify a particular one. So we have taken PLK1. What it is? It contains 603 amino acids. Look, its molecular weight, its heaviness, its structure. Okay. Now, so how many amino acids are there? 30. Existing? 30. Amino acids कितने आते हैं? 30. 52. 32. 32. किसी 20 आते हैं? मी कन्फ्यूज केलं पण मला 20 वाटतं माहिती आहे तो काय करतो हे एमसीक्यूज मध्ये प्रॉब्लेम हे ABCD दिलं की नंतर असं वाटेल पण 603 तर नसतील sure 603 amino acids no 20 amino acids are there. So this particular PLK1 contains 603 amino acids. Okay. Now, once this target is identified, what we have to do is we have to get this target structure. How to get the structure of this target? Where it is available? It is available on protein data bank. Protein data bank is there. A known site, RCSB site is there, where the structures of proteins are available. Those structures who have been already elucidated, these protein structures are available. Suppose someone identify a totally new protein, new target, then you have to go for its IR, NMR, 3D NMR, then X-ray crystallography, every studies, and then you have to establish its structure, chemical structure. If totally new target is identified. But normally at this stage, we work on the available protein data bank. So this is PLK1, it's available on the protein data bank. So you can get that structure from protein data bank. This is computational data. So in the software you can import that structure of PLK1, which contains 603 amino acids. Okay. Now this structure is known. Now we have lock. Now out of this protein structure, you have to identify what is its binding site. The lock is keyhole identifier. Right? So out of that 603 amino acids, which 5, 6 or 10 amino acids, those who are involved in binding with the ligand, that is needs to be identified. Okay, this is called as identification of binding site. Okay, once binding site is identified, you have to design a library of compounds. Because we have to do a virtual screening. Virtual screening means you don't have to synthesize all. The computer is doing a simulation. Okay, so you can design a library maybe of 10 compounds, 100, 10,000, 1 lakh, or whatever. So you can design that virtual library and go for docking studies, where you will find that which is best fit in that. Then accordingly you can shortlist and go for synthesis. This is all the overall project is. Next. Okay. Please. Now I hope those who want to work in this particular area, they have understood how to plan your research in this area. Identify your disease in which you want to work. Then study the pathophysiology of that disease properly. Identify target. Then identify ligand if available from literature. Then design a virtual library. Go for virtual screening of multiple compounds. Shortlist selective compounds for synthesis. Synthesize it. Structural elucidation of that compound. Biological activity and further. This is how the steps can be done in this. Okay, next. See, this is actually, if suppose anybody wants to apply for funds, those who are providing research grants, the list is given. It may not be right now useful for you. Next. Some international agencies are also there. They also can provide grants for research projects. So, okay. So, selection of project. When you want to select project, it should be novel. Novel means what? Novel. Nobody has done earlier. So you have to check its novelty first. Anywhere there are sources, various sources available, databases are available. From that databases, you have to confirm first that whatever you are going to do, it is novel. Your project should be on current issues. Whatever latest, when COVID was there, everybody focused on COVID. Like that. When current issue, you have to focus on current issue. Then practical feasibility, of course. Just thinking of no use. It should be practically implementable. Useful to society. That is important. Again, cost effective. Suppose you are giving something, but if it is not affordable, no use. So cost effectiveness is required. Commercially viable. Commercially viable means what? जेवढा खर्च घातला तेवढा तरी यायला पाहिजे किंवा प्रॉफिट त्यात मिळालं पाहिजे. समजा ऍटलीस्ट तेवढं ब्रेक इव्हन असलं तरी ठीक आहे. कमीत कमी लॉस मध्ये जरी म्हणजे घर घालून काय म्हणतो तसं नाही चालत. घर घालून ड्रग डेव्हलपमेंट आपण असं नाही करत. काही वर्ष मी केलं आपण क्रिएटिंग एम्प्लॉयमेंट. Creating employment means what? Suppose you are doing some project, it should create some employment to the others. Right? So, suppose you set up some manufacturing, or suppose you identify or develop some drug and manufacture it. You are giving employment to many manufacturers, sellers, everything. That is again great. Okay, next. Now, okay, one more example. That drug resistance we were talking about, that I would like to share with you. See, I earlier told about sulfonamides. Sulfonamides because they are not in use. Sulfonamides, they are competitive inhibitors of PABA. Right? And so the folic acid photosynthesis and folic biosynthesis, that process is affected. Now, where this is a compound which we have developed, they are acting on the same pathway, but at different sites. The tannin binding pocket, you can see this. Two pockets are there, PABA binding and tannin binding. A lock with two keywords. So we targeted another tannin binding pocket, so that the same pathway can be inhibited. Got it? So these are the different molecules. Means this is novelty, you can say. So known thing. DHPS inhibitors, sulfonamide class of drugs, they are so known. But what novelty we found is the different target is different. Okay, next. Okay, sorry. Next. So, when you have to prepare any research proposal, what should be the component? Title, objectives, introduction, background literature survey, methodology, time frame, and the availability and budget. Okay, next. Title. Title should be unique, punchline. Look, the example is given. The title can be written like this. See, synthesis and molecular modeling studies of natural derivatives as polo-like kinase inhibitors and anti-cancer agent against breast cancer. This is one type of title. Read the second title, which is promising. Second, targeting PLK to design, right? So this is how target should be some specific and like a punchline. Next. Once target title is set, you have to set its objective. Objective means what? आपल्याला काय करायचं? हे करायचं, कशासाठी करायचं? Okay, you have to set objective. Next. Introduction. What is there in the introduction? It shows a need of that work. का करायचं? That should be clear in this introduction part. That is called a need of research. The need of research should be clear. Okay, next. When need is clear, you look at the background. Means literature survey. Anti-cancer करायचं, PLP इनहिबिटर करायचं आहे. ओके, मला ते करायचं आहे. पण आधी पण काय केलंय, त्याचे बॅकग्राऊंड काय आहे, मागे 10 वर्ष 15 वर्ष 20 वर्षात काय 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 काम झालंय ते सगळं बघणं म्हणजे त्याची लिटरेचर सर्वे बॅकग्राऊंड गेटिंग. काही कुठलाही आपण करायचं ठरवलं तर आपण काय आधी आपल्याला कुठल्या गावात जायचं असेल नवीन ठिकाणी तरी आपण कोण जाऊन आलं, कसं गेलं, कुठला रूट होतं, काय वापरलं, बस गेलं, ट्रेन गेली, फ्लाईट गेलं, काय काय, दॅट इज बॅकग्राऊंड. Okay, so this is background. Okay, next. Methodology. Now you have decided what you have to do. So how to do it? Experimentation part, method. By which method you are doing? That methodology has to be set. Okay. So when you are designing methodology, see that each and every claim which you make, that should be experimentally supported. Accordingly, you have to set a methodology. Next. Of course, time structure, how much time is required. Every project, Pharm, say 2 years duration, PhD, maybe 3-4 years duration. Any project, suppose any funding agency is giving a project, it has some duration, 2 years project, 3 years project. So timeline is everywhere. So you have to follow the time frame and you have to set first. This is for this purpose, this many days, this purpose, this many days, like that. The time frame needs to be framed. Next. Then you have to check where you are working, where you want to perform this, what are the facilities available. Right? So whether these facilities are sufficient to satisfy your aim or your objectives, or to achieve your objectives. Like that. So the facilities, if they are not available, you have to create them. Or you can ask for that facilities. Those if there is someone sponsoring. Next. Okay, this is for particularly for the project funding. Suppose you need principal investigator, co-investigator, some supporting staff. These salaries can be covered in this, right? Suppose those who are funding any projects, they funds for everything. They funds for small things purchase to manpower. Everything they provide, everything, but you have to give budget. Right? Okay, next. Yeah, budget. Budget is, see how much is required for staff. Suppose you need 2 or 3 staff. The salary of staff for say 3 years, that you have to calculate. Non-recurring is what? Equipments. Any fixed asset. Suppose you are going to purchase equipments. That is non-recurring. Recurring means chemicals, glassware, that can be. Then travel. Suppose you need any travel. Contingency, emergency, suppose anything is required. And any more HR. So accordingly you have to frame budget. You have to ask for money. And if they are agreeing, if they found it is good or something outcome based, they may provide you funds. Okay, next. Now, see, suppose you do some research. Suppose you do some research and you want that it's your intellectual property and you want to protect it. So what to do? You suppose you want to put some last 2 slides. Suppose you want to protect your property. Then what to do? People do research. One thing what they used to do is they publish it or present it somewhere. Publish it means what? You can submit to a journal. Early morning, what I was talking like journals, elsewhere, Springer, Willy, these are the publishers. They publish different journals like Journal of Medicinal Chemistry, Biological Medicinal Chemistry, different journals. So they used to publish these journals, scientific journals. What you can do? You can submit your manuscript as per their format to that publisher. They review it with experts. Suppose they find it is suitable but needs some corrections, needs some revision to be done. They communicate it that these are the revisions you have to make and resubmit. Then if it is found suitable and as per their guidelines, they will accept it and they will publish in their okay, research you have done and published, finished. Then people will refer it. It will be a guideline for new researchers. What someone has done, what he has done. Like literature survey you will appear. He has done this, he has done this. So it will give some directions. But it will not remain your own intellectual property. That is what is happening with publication. Getting it? So it is a public, or say it may. Hello. It may be a public property, or that it may be a right of that publisher, because you are giving copyright to that agreement we used to give them. So the whole right, the author is giving whole right to publish. So apply that. But if you want to protect your research, if you want to protect your property, intellectual property on your own, you can go for patent. But if patent, suppose you do something, first you have to check whether it is novel or not. Now it is checked. Navin okay, I think you can go for patent. If it is not novel, forget it. Then if it is novel, yes, you draft patent. It may be provisional patent or complete patent, whatever. You file that patent. Then after some period, it will be published into the patent gazette. Then you can apply for examination of that patent, so that the patent office will appoint some examiners. They will examine your patent, whether it is really novel, whether it is really of something else. Whatever claims you have given in the patent, whether these are valid claims. If it is so, then they will grant it. Or otherwise, before granting, if they found some queries, examiners find that there are some issues or if they have some doubts, they will raise that queries. So you can reply to that queries. If they satisfy, then they grant it. Now the patent is granted. Now you are the owner. But it is for 20 years. But you have to maintain it. Again you have to pay some maintenance charges. So both for 20 years you have to maintain it. So this is all about Indian patent. But even if you feel that your research needs to be protected across the globe, so you can go for world patent or US patent like that. So accordingly, your intellectual property can be protected by patents. Okay. So publication and patent. This is a major difference between. Okay. Next. So suppose you go for patent, likewise these are the different benefits after patent. It gives a monopoly, right? Then royalty you may receive, like royalty, global recognition, goodwill, many things. Still, nobody will call honorable. Nobody will call. Suppose you want to be honorable, you fill your form in the next. Don't fill that patent file. Go for that there. Okay. Thank you. I hope you listen.
Now there is some questions. No questions? Or else you can take the mail ID of sir in the.
See my mail ID.
Don't have any doubts right now. Later also you can approach sir.
तुम्ही म्हणजे कॉलेजवर येऊन पण भेटू शकता. तुम्हाला वाटलं तर तुम्ही पिक्चरच्या माध्यमातून काय विचारायचं तरी विचारू शकता. तुम्हाला स्वतः पर्सनल काय असेल
Don't have any. Let us not have any hypothetical barriers. Hypothetical barriers in the sense this is one college, that is other college. We are all under one roof, that is Savitribai Phule University. Sir is a very renowned professor from 6PPU. Recognized in the entire university.
तुम्ही कॉलेजवर सोडा, तुम्ही मला भेटायला येऊ शकता.
So you can ask any questions. Yeah, please.
केलेलं नाही असं नाही. काय बघितलं तुम्ही? कुठले ग्रीन सिंथेसिसचं बघितलं? कशाने बनवलं? ग्रीन सिंथेसिस मध्ये काय म्हणतात? ग्रीन सिंथेसिसच्या भरपूर मेथड्स आहेत. म्हणजे अगदी मायक्रोवेव्ह सुद्धा ग्रीन मध्ये. ग्रीन म्हणजे काय? एन्व्हायरमेंटल फ्रेंडली. एनिथिंग विच इज एन्व्हायरमेंटल फ्रेंडली इज ग्रीन सिंथेसिस. कुठल्याही मेथड वापरू शकता. असं आहे तुम्ही अगदी ज्या मेथडमध्ये सॉल्व्हंट वापरून करताय तीच मेथड तुम्ही विदाउट ए सॉल्व्हंट तुम्ही ड्राय फ्युजनने जरी केली तरी ती ग्रीनमध्ये म्हणजे कंपॅरेटिव्हली ग्रीनमध्ये पडतो. असं आहे. त्यामुळं चॅलेंजेस काहीच नाही. तुम्ही फक्त वेगळं काय करताय का ते बघायचं. तर कसं आहे माहिती का, एनफॉर्मचा प्रोजेक्ट आपण त्याच्यातलं काय बघतोय. माझ्या दृष्टीने एनफॉर्मचा प्रोजेक्ट हा फक्त शिकण्यासाठी आहे. तुम्ही जर म्हणता सर मी एनफॉर्मच्या प्रोजेक्टमध्ये एखादं पेटंट करेन, आम्ही कमर्शियल प्रोडक्ट डेव्हलप करेन किंवा काय, ते तेवढ्याशा 6 महिन्या 1 वर्षाच्या काळात ते पॉसिबल होत नाही. फक्त ही सगळी प्रोसेस आहे. ह्याच्यात तुम्हाला रिसर्च मेथडॉलॉजी शिकणं एवढंच तुमचं ध्येय हे असू दे. पण प्रोजेक्ट असा निवडा की जर तुम्ही पुढे त्याला एक्सटेंड केलं तर काहीतरी देत राहील. तुम्हाला जर ह्या वर्षाच्या 2 वर्षाच्या काळात असं वाटत असेल की आपण काहीतरी कमर्शियल आउटकम निघू शकेल तर ते इतकं क्विक व्हायला फारसा अवघड आहे. तेवढ्या वेळेला नसतो आपण. पण असा प्रोजेक्ट निवडायचा की जर पुढे जाऊन तुम्ही रिसर्च मध्ये राहिला किंवा तुम्ही पुढे काय करा कुठल्याही फील्डमध्ये जा तर त्याला जर पुढे कंटिन्यू केलं तर काहीतरी करता येऊ शकतं. म्हणजे इथं फक्त तुम्ही आधी एनफॉर्मचं प्रोजेक्ट करिक्युलम मध्ये डिझाईन करण्यामागचाच उद्देश जो आहे. रिसर्च काय असतो आणि कसा करावा हे शिकणं, ही प्रोसेस शिकणं एवढं जरी झालं ना प्रोजेक्टमध्ये तर ठीक.
Very good. What is your name? Please learn from her.
एक मुळात काय म्हणते का तुम्ही पहिलं बोलायला शिका. सगळ्या शिकण्याच्याच प्रोसेस आहे.
At least ask some questions. That is what we are doing. We are not expecting you to give the answers for the questions.
सुरुवातीला तू तेच म्हटलं की आपल्याला चुकीचं बोलायचं पण धाडस पाहिजे. ते पण उदाहरण त्याचं काय वाटलं नाही पाहिजे. सगळे हसले तरी काय नाही वाटलं. कसं म्हणतो
रिव्ह्यू पेपर किंवा रिसर्च आर्टिकल बनवताना AI वापरावं की नाही?
म्हणजे आपण ते कशासाठी वापरतो हे म्हणतो. आपल्याला म्हणजे थोडा असिस्टन्स म्हणून वापरत असतो तर ठीक आहे. काही तुमचं रिपिटेटिव्ह वर्कला जर तुम्हाला सपोर्ट म्हणून वापरत असतो तर आपण काही डॉंकी वर्क करणार नाही. पण तुम्ही तेवढं त्याच्यात ते जसं तसं तेच घेत असाल. आपण आपलं आय वापरतच नाही इंटेलिजन्स. दुसऱ्याच ते आय वापरतो काय करतो आपलं इंटेलिजन्स वापरतो. आर्टिफिशियल घ्या पण आपलं ओरिजिनल इंटेलिजन्स पण त्याच्यात वापरत असतो तर त्याला काय हरकत नाही. तसं नाही तुम्ही सपोर्ट घेऊ शकता पण तो स्वतःचा नॅचरल इंटेलिजन्स वापरायलाच लागतो. मग इथं काय होतं नॉर्मली AI मध्ये जसं तसं ते जे आहे ते तसं आपण त्याला काही बघत नाही. म्हणजे आता आम्ही वापरतो पण फक्त एक बॅकअप त्याला प्रॉम्प्टिंग पण म्हणतात. ते आपलं ओरिजिनल प्रॉम्प्टिंग आहे. आपण प्रॉम्प्टिंगला काय द्यावं ते पण यायला विचारतो. दुसऱ्याला विचारतो म्हणजे दुसऱ्या याला प्रॉब्लेम काय इथं प्रॉब्लेम. अदरवाईज टेक्नॉलॉजी कुठली वापरू नये असं मी म्हणतो. टेक्नॉलॉजी ती वापरायलाच पाहिजे फक्त कशासाठी वापरतोय ते म्हणतो. नो प्लीज हे चर्चा आहे.
You can use AI for citation. Whatever the repeated citations which you like that they make at the end of the paper. It will be much like comma, that thing. If you can give it to the AI, real agent, and give it.
Right.
सर जर आपण 2 गटचं मेथड डेव्हलपमेंट करतोय कॉम्बिनेशन करू बट सिंथेसिस नाही करू शकत कारण ते 2 API आहेत. तर ते आपण बॅलन्स कसं करणार म्हणजे जर सिंथेसिस एका गटाचं केलं
मेथड डेव्हलपमेंट हा वेगळा विषय आहे आणि सिंथेसिस हा वेगळा विषय आहे. दोन्ही पण पण तसं काही नाही. मग तिथं तुम्ही तुम्ही 2 केमिकल सिंथेसाइज करा वेगळे, ते ड्रग असतील असं नाही आणि त्याचं सायमल्टेनियस करा.
नाही, बट मी ड्रग चूज केले.
ड्रग चूज केले असतील तर त्याचं सिंथेसिस तुम्ही ड्रगचं सिंथेसिस करून ऑलरेडी सिंथेसिस एस्टॅब्लिश आहे ना, तर ते नाही होणार.
ते कॉम्बिनेशनमध्ये मेथड डेव्हलपमेंट नाही झाली.
तिचं म्हणजे फॉर्म्युलेशन करायचं त्या 2 ड्रगचं आणि नंतर मग ते मेथड डेव्हलपमेंटसाठी जाऊ शकतो.
त्याला दोन्ही 2 ड्रग आहे, मग तुम्ही त्याचं फॉर्म्युलेशन करणार, त्या सिंथेसिस करणार, 2 वेगळ्या गोष्टी
कर सकते हैं. There are functional groups which can react with each other. उसका hybridization
नाही त्यांना म्युच्युअल ड्रग करायचं, म्युच्युअल ड्रग करू शकतो, म्युच्युअल ड्रग्स करू शकतात. 2 ड्रगचं एक कंपाऊंड बनवलं म्युच्युअल ड्रग म्हणतात, दॅट इज म्युच्युअल ड्रग. म्युच्युअल ड्रग करताय मग आणि मग ते तुम्ही हायड्रोलाइज करा, 2 ड्रग फॉर्म होतील आणि त्याचे एस्टॅब्लिश करायचं मेथड डेव्हलप. म्हणजे कसं एक ऍसिड आणि अल्कोहोल असेल ड्रग फॉर्म त्याचं एस्टर बनतो, तो म्युच्युअल ड्रग असतो आणि जो बॉडीमध्ये जातो तेव्हा हायड्रोलाइज एन्झाईममध्ये तो ब्रेक होतो आणि 2 ड्रग वेगळे होतात आणि त्यांचे मेकॅनिझम तुम्ही काम करता. तसे तुम्ही म्युच्युअल ड्रग्स करू शकता.
Any other? Shall we wind up? Precious time you spend with us, and we are so much grateful to you for the wonderful sessions which you have delivered in the morning as well as in the afternoon sessions. And the kind of response that you can see from the students itself shows that your presentation was commendable and excellent. And all faculty also, I told all faculty to listen to your presentation, leaving from their particles. And once again, on behalf of our management, we would like to extend a heartfelt thanks to you for presenting these students. Thank you. The details of solubility of drugs, right? Why solubility is determined and why the concept of solubility we are studying in pharmacy. What is its relevance? What is its application? Where exactly this concept is going to be useful for you? Those things, let us have a discussion in the next couple of slides. Next slide, please. So every lecture should have some outcomes. Right? Outcome in the sense, whatever the 1 hour or half an hour, whatever the discussion that we are going to have, should be focused. It should not be something zigzag. It should not be something like whatever that comes into our mind, let us discuss. It should not be like that. Learning outcome is, it should be focused, and whatever that we are going to teach, it should be delivered to the students. That's what the lecture outcomes is all about, right? So after this lecture, you should be able to define and classify solubility of drugs, which means at the end of this lecture, if I ask you a question, define solubility means you should be in a position to define the solubility and classify. Classify means classification of different types of solubility is also there. That also let us have a discussion. Then explain factors affecting solubility. What are the different factors which actually influence the solubility? Which factor increases the solubility? Which factor decreases the solubility? That also let us discuss, and by the end of this lecture we should be able to tell all the factors that influences solubility. Next, relate solubility with bioavailability and formulation. This is the ultimate goal, right? Step by step we are coming. The lecture outcomes are step by step. In the first step, the definition, classification. In the second step, you are going to find out, okay? So you are going to find out the factors that affect the solubility, explain factors that are affecting the solubility. Then relating that solubility with then what is bioavailability? That also let us discuss in the coming slide. Next. So objective is understand basic solubility concepts, study physicochemical factors, learn solubility enhancement techniques. These are the lecture objectives. Next. So now, as just now I told that why solubility is matter. Why solubility matters. Right? Solubility. First look at the word itself. Solubility, which means solutes ability. It's a combination of the word solute plus ability. The ability of that particular solute to dissolve in a particular solvent. Right? So when solute, which means the solid is solid and liquid is solid. And how this solid and liquid are interacting with each other. right? So it's a kind of solubility means it's a study of the interaction between solute and solvent. Whether the interaction is fruitful interaction, if the interaction is feasible, possible, then solubility occurs. If there is no interaction, if there is no affinity, so for a solubility to occur, there should be some affinity between solute and solvent. If there is no affinity, then there is no interaction. If there is no interaction, there is no solubility. In the solubility process, what happens? Right? First, the solute comes in contact with the solvent. Solute comes in contact with the solvent. It means solute is in solid state, solvent is in liquid state. And you know interfacial phenomena. Right? In the interfacial phenomena, you know very well about the different types of boundaries. Right? Solid-solid boundary, right? Solid-liquid boundary, right? So they are called interface. Right? Surface and interface. Now here, what is the interface? Solid and liquid interface. Now this solid must be wetted by the liquid. Otherwise, solid will not enter into the solvent. That is liquid. Right? There will not be any type of interaction. So these things you should keep in your mind. And now, drug solubility means drug must dissolve before absorption. Poor solubility, poor viability, critical in oral dosage forms, which means, so solubility, just by looking at the solubility, we can tell whether that drug is going to absorb or not. Suppose you have a drug, right? For the drug to elicit its biological action or pharmacological action, first it has to dissolve in your stomach fluids. That is, in your stomach fluid means that is acidic environment which is present in your stomach, that is acid, and also in the intestines you have basic environment. So in this it has to, which means in the body fluids, if it is taken orally, it should dissolve in your stomach acid. If it is taken in the IV route, it should dissolve in your blood, right? So it means in your body fluids, whatever the drug that you administer, it should dissolve. If it does not dissolve, then it cannot produce any therapeutic action or pharmacological action or biological action, whatever you call. Which means for all types of actions or for a drug to elicit its action, the first and foremost beginning step is solubility. If it does not become soluble, it cannot be absorbed and it cannot be reached to the site. In the pharmacology you study about this. Drug has to reach the site of action. In which form? In the molecular form. In the molecular form means in the dissolved state. In the dissolved state means, you know, you might have already studied about dispersions. You might have studied about dispersions. Have you studied or not? Dispersions. What is dispersion? What is a dispersion? Dispersions are biphasic systems. Right? Disperse phase, dispersion medium. Suspensions, emulsions, they are the examples of dispersions. What is the difference between a solution and a dispersion? Solution, the solution is a homogeneous mixture. Dispersion is a heterogeneous mixture. And in the solution you have solute plus solvent. And in the dispersion you have dispersion medium and dispersive phase. Right? And in the solution, what is solute? The one which is in low quantity. Right? That is called in less number of parts. That is called solute. And the one which is in larger quantity is called solvent. Here, dispersed phase also, the one which is distributed into the dispersion phase is called dispersed phase. And the only difference is, one which is soluble, it becomes solution. If it is insoluble, then it becomes dispersion. Right? So, which means it is the particle size which is going to decide whether it is going to become a solution or a dispersion. Right? If the particle size of the dispersed phase, that is solute, is less than 1 nanometer, then it can be considered as a solution. For that matter of fact, you can also call solution as molecular dispersion. Solution is also a molecular dispersion, which means we are distributing solutes into the solvent. Solutes into the solvent we are dispersing, right? So by doing that, what is happening? Solution is going to be formed, right? In case of dispersion, so dispersion phase is distributed into the dispersion medium and its particle size is greater than 1 nanometer. In between solution and dispersion, you have one intermediate colloidal solutions. Right? Colloidal dispersions. You might have studied about this. Colloids. Colloids have the property of solution as well as dispersion. Partially soluble. Partially insoluble. Based on that, you classify once again colloids into 3 different types. Lyophilic, lyophobic, and another one is the association colloids. Lyophilic means affinity towards solvent. Lyophobic means hating towards the solvent. Association means having both groups, hydrophilic and hydrophilic surfactant molecules. Right? Now, here coming to the definition, solubility is the prerequisite step before drug absorption. Poorly soluble drug cannot be absorbed, which means the best example is griseofulvin shows erratic exam. Absorption of griseofulvin is very erratic. What is griseofulvin? In which type of condition it is used? Griseofulvin. Have you heard of this particular thing? Antifungal. It is an antifungal agent having very, very low solubility, very poor solubility, and this drug has been tried during the COVID infection. Have you heard of the term called mucormycosis? Mucormycosis. In mucormycosis, this was the drug of choice, griseofulvin. Right? Next. Now coming to the solubility definition, because I told in the lecture outcomes that at the end of this particular lecture, first lecture outcome is we must be able to define solubility. Second, classify solubility. Third, explain factors affecting solubility. And the fourth one, relate solubility with bioavailability in the formulation. These are our objectives and outcomes. Right? Now coming to the definition, maximum amount of solute dissolving in solvent at standard temperature and pressure is called solubility. Which means if you take one particular solute, right, and add it to the solvent, right, so you should go on adding that solute to that particular definite amount of solvent. Here one thing is missing. Maximum amount of solute dissolving in solvent at a specified temperature is, here solvent is restricted, right? The definite amount of solvent, right? Maximum amount of the solute that can be dissolved in the definite amount of solvent at a definite temperature and pressure. Which means quantity of the solvent, temperature, pressure, these are the variables which are going to influence the amount of the drug that is going to become soluble in the solvent. Which means if you want to define the solubility, maximum amount of the drug that can be soluble in a given solvent, that is very important, in a given solvent at a specified temperature and pressure. This is the actual definition of the solubility, right? And another terms if you can use, it's nothing but maximum means concentration. It's nothing but concentration. Concentration of the solute. Concentration of the solute in a saturated solution. There are 3 types of solution once again: unsaturated, saturated, and supersaturated. If you want to define the solubility in another way, right, the concentration of a solute in the saturated solution can be called as solubility at a definite temperature and pressure. If you are saturating means, what does it mean? Saturating means, what does it mean? There is an equilibrium. Saturation means there is an equilibrium. What is that equilibrium? Between the dissolved and undissolved amount of the solid or solute which is present in the solution. Which means in the solubility process, it's a kind of mass transfer. Mass transfer in the sense solid state is changed into liquid state. Right? Solid state of the solid is changed into liquid state until and unless the concentration of solid, dissolved and undissolved becomes equilibrium. At that particular point, when there exists an equilibrium between dissolved amount and undissolved amount, then there is no longer be a mass transfer. Right? Because the solution is saturated. The ability of the solvent molecules to surround solute molecules is over. students. 100 students means its saturated capacity is 100 students. If 110 students come, it cannot occupy because all the benches are occupied. Similarly, in the saturated solution, all solvent pockets are occupied with the solutes. Hence, even if you add some amount of this solute, it cannot enter into the solution because there is a saturation. In the unsaturated solution, if you take the concentration, that cannot be called as a solubility because some more amount is going to be added. It can receive some more amount. That's why if the concentration, if you measure the concentration of the unsaturated solution, then it cannot be called as a solubility. Why? Because the definition of the solubility itself says that the maximum amount of the solute dissolving in solvent, the given solvent. In the unsaturated solution, it's not a maximum amount of the solute because some more amount of the solute you can add. But when it comes to saturated, no more amount can be added. And when it comes to supersaturation, if you can heat, if you can increase the temperature, some more amount of the solute can be accommodated in the solvent. That is called supersaturated solution. And once the temperature is withdrawn, once the temperature is decreased, then the excess amount of the solute that is dissolved into the solvent can precipitate, can come out as a crystals. This is used as one of the crystallization techniques. To form the crystals, people are going to use supersaturated solutions. First they prepare the supersaturated solution, immediately they decrease the temperature. The moment they decrease the temperature, the excess amount of the solid that is dissolved in the solvent will crystallize as precipitate. Right? It has its own use, I will tell that later. Right? Now, so example, if you can take 1 gram of sodium chloride dissolving in 2.8 ml of water at 25 degrees centigrade is called the solution. Which means if you want to explain the solubility of sodium chloride, 1 gram, this is the IP standard and USP standard, IP and USP, Indian Pharmacopoeia and United States Pharmacopoeia. If you can refer those standard texts, how solubility is referred and mentioned, it is mentioned in terms of grams. 1 gram of so-and-so substance will dissolve in how many ml of so-and-so solvent. Right? So here, the same expression you can see here, 1 gram of sodium chloride dissolving in 2.8 ml of water and the temperature is very, very important, that is 25 degrees centigrade. When there is no mention about pressure, it is understood that it is atmospheric pressure. You may ask me here, pressure is not mentioned. Whenever there is no mention about pressure, it means it is understood that it is atmospheric pressure. What is the atmospheric or atmospheric pressure? Next. Then, what are different types of solubility? Now we understood about the definition of solubility. Solubility means maximum amount of the solute which can be dissolved in the given solvent at the specific temperature and pressure. That's it. This is called solubility. Another definition is, in case of saturated solution, the concentration of the saturated solution at a definite temperature and pressure is also called solubility. Right? And if you want another definition, the interaction between solute and solvent. Solubility is all about the interaction between the solute and study of interaction between solute and solvent is nothing but solubility. Now here, types of solubility, if you can refer to here, intrinsic solubility is one type, apparent solubility is another type, and kinetic solubility is another. So there are 3 different types of solubilities which are used in our pharmaceutical sciences. We quite often refer to these particular solubilities, right? What is intrinsic solubility, what is apparent solubility, what is kinetic solubility, right? So which means intrinsic solubility means what? Intrinsic means the unionized. Once again, you might have studied about ionized state and unionized state, right? This is once again the concept which comes in dissociation constant. You might have studied about dissociation constant and association constant, right? Do you know about this dissociation and association? Dissociation means how the solute dissociates in the polar solvent. So dissociation is always a process which takes place in the polar solvents. Association takes place in the non-polar solvents. Right? When one molecule is divided into 2 parts, then it is called dissociation. ionized and unionized parts, right? So it happens because of the polarity, right? So intrinsic dissolution rate refers to the solubility of unionized solid, right? You can see unionized drug solubility. Solubility refers to the solubility of unionized drug, right? Once again, solubility of ionized drug, unionized drug is very, very important. Generally, if you refer, the solubility is the total amount of the ionized plus unionized fraction of the drug, right? A drug at a particular pH exists in two forms, ionized form and unionized form. If you refer to the both forms, right, total amount of ionized and unionized form which is dissolved in particular solvent, then if you can refer to that, it is called solubility. But if you specifically mention about the unionized amount of the drug that is soluble in a particular solvent, then it is called intrinsic solubility. Why here only unionized amount of the drug solubility is given importance? This you have to understand. Why? Because as per pH partition theory, the ionized amount of the drug is not going to be absorbed. Why? The purpose of studying solubility is to predict the absorption behavior, right? Suppose one new drug is being given to you, and now you want to check whether it is going to absorb into body or not. The simple test that you can perform is take it and add it to water. Add it to 0.1 normal HCl. Add it to blood pH, which means 7.5, 7.4 is the blood pH. Buffer of 7.4 pH. Buffer of 6.8 pH. To these liquids, if you add that particular solvent and check whether it is soluble or not, and primarily you can predict whether it is going to be absorbed or not, because the amount of the drug that is soluble in the body pH reflects the amount of the drug that is going to be absorbed, right? So in this particular, the purpose is to study and predict the absorption behavior of a particular drug by studying the solubility, right? And here in absorption, unionized portion of the drug is more important than ionized portion, right? Even if the drug is soluble, if the drug is having more ionized portion and less unionized portion, then it cannot reflect the true absorption behavior. Suppose you have taken one drug and that drug absorption is very high and you felt very happy. Yes, my drug is going to be absorbed, and you tested it in the in vivo system, in the animals or in, whatever the instruments animals that you use. In vivo system you have tested it, let us say. Later on you found that nothing is absorbed. Right? Actually, if the drug is more soluble, it has to be absorbed. But after conducting the experiment, you came to know that nothing is absorbed, because the total amount of the drug that was soluble is only ionized portion but not unionized portion. If unionized portion is present, then the drug can be absorbed. That's why intrinsic solubility is the true parameter which can predict the absorption based on the solubility. That's why, so if you determine intrinsic solubility, that can serve as an indicator to assess the absorption of a particular drug, right? Next, kinetic solubility, apparent solubility. Apparent solubility means it's not constant. The solubility of the particular drug is not constant. It varies from pH to pH. So this type of drugs are nothing but they are either weakly acids or weakly bases. Weak bases and weak acids. If your drug is a weak base, if your drug is a weak acid, then the drug is going to have pH dependent solubility. That is called apparent solubility. Suppose weak acids you take. Weak acids have more solubility in basic environment, because acid base reacts and then solubility increases. And weak bases will have more solubility in acidic environment. Right? When it goes to basic environment, its solubility decreases. When it enters into the acidic environment, so your gastrointestinal system consists of different types of pH environments. In stomach you have one pH, in intestines you have another pH, large intestines alkaline pH, and colon too much alkaline. Right? So your absorption depends on solubility. Suppose weakly acidic drug is there, it is going to be absorbed very well in the intestines, because weak acids dissolve in the basic environment. If your drug is weakly basic, then it is expected to be absorbed in the stomach more, because in the stomach we have acidic environment. Right? So that is how apparent solubility is used to predict. The pH dependent solubility. If you vary the pH, so far you came to know, you only know that solvent, right, and temperature, pressure, these are the three variables which influences the solubility. Now another point is added, that is pH. If you change the pH, then also solubility. My dear students, please remember, if you can understand these variables which influences solubility, then you can easily interpret and guess factors that influence solubility. Now the same variable becomes the factor. Same variable temperature will now influence the solubility. Same pressure influences the solubility. Same pH influences the solubility. Right? So variables that influences solubility, if you can understand, and amount of solvent also influences the solubility. Now next, kinetic supersaturated solubility. Supersaturated solubility is called kinetic solubility. Why kinetic? Why the word kinetic is being mentioned, used here? Because when you heat, when you increase the temperature, what will happen? Kinetic energy increases. Because the kinetic energy increases, molecules tend to be in a motion, random motion. As a result, the solubility is going to be increases because they get some energy, because of which they will be able to get solubilized. The excess amount is going to be. So kinetic solubility refers to supersaturated solubility. So what are three different types of solubility? First one is intrinsic solubility. Second one is apparent solubility. Third one is kinetic solubility. And definition of solubility is maximum amount of the solid which can be dissolved in the given solvent at a definite temperature and pressure. Now first lecture outcome is over. Done or not? Solubility definition and classification, types of solubility. Next. Then comes expression of solubility. Right? How will you express? There are different methods. Concentration is one method to express the solubility. Right? So parts of solid required for one part of solubility is one expression. Right? That solubility table which you can see in P.P. Martin, Physical Pharmacy written by Martin textbook. How many of you have referred to that particular text? Physical Pharmacy by Martin, which is there in the library. Have you gone through it? At least have you seen its cover page? Very, very important book. Please refer to it. You will get good knowledge when you refer and read to that particular book. Physical Pharmacy by Alfred Martin. In that book, there is one table. The table is expression, solubility expressions. Right? I can challenge you. In every GPAT examination, one question will be from that particular table. So it defines terms like soluble, poorly soluble, partially soluble. When we can say that a particular solid is soluble, when we can say that a particular solid is insoluble. Right? When you can say. Suppose you have taken one particular solid, whether it is soluble or insoluble, how to differentiate it, how to define it. Just by taking in the selected, suitable, specific or given solvent, just after adding it, since it is not dissolved, it is not solvent. No. If you increase the solvent ratio, if you increase the solvent volume, then solubility may increase, right? So, which means up to how much level you can raise the volume of the solvent to check whether it is completely soluble or insoluble. That is given by that particular table. Poorly soluble, partially soluble, soluble, sparingly soluble, very soluble, slightly soluble, last insoluble. Such expressions, and those expressions are defined based on the term called number of parts of solvents required for one part of solute. If less than one part of solvent is required to solubilize one part of solute, then it is called very soluble. Very soluble. Which means 1:1 is not that. If one part of solute is dissolving less than one part of the solute, then it is highly soluble, very soluble. When you can say a particular drug is soluble, as not not drug here, we must use substance. When a particular solid or solute is said to be soluble, when one part of solute dissolves in 30 parts of solvent, then you can say that that particular drug is soluble. Right? If it is dissolved in less than 1 part, you can say that very soluble. If 1 part of solute, right, does not dissolve even in 10,000 parts of the solvent, then you can call it as insoluble. Right? This is how the expressions, solubility expressions are given, and solubility expressions are recalled, remembered, referred. Right? Next, molarity and molality. This is concentration terms. Right? Concentration, because as per the definition of solubility, concentration of the concentration of the saturated solution is nothing but soluble. Solubility is defined as the concentration of the saturated solution. Then concentration, how to measure? You have molarity, you have percentages, right? Weight by volume, volume by volume. So many expressions, weight by volume, weight by weight, and percentage expressions are also available. Okay? First one is number of parts versus number of parts of solute versus number of parts of solvent. Second one is molarity and molality. Third one is percentages, weight by volume, volume by volume, or weight by weight. And this is how you can express the solubility. So solubility can be expressed quantitatively using standard units. So paracetamol solubility is 1 part in 70 parts of water. So it is slightly soluble, which means paracetamol is slightly or sparingly soluble. Because what I said, the definition is, if you have to call something as soluble, 1 part should be soluble in 30 parts. Here 70 parts, which means slightly or sparingly soluble. Next. So importance, what is the importance of solubility in pharmacy? So affects absorption rate, so which means solubility, if you can study, right? So that solubility you can use to predict the absorption rate, right? Next, determine dosage form. So which dosage form you have to use, right? So if it is, depending upon the solubility only, we have to use whether it should be used as tablet dosage form, capsule dosage form, or injection. Okay, so it all depends upon the solubility. So if it is practically insoluble, then you can go for suspension also. You have an option. Suspensions you are studying. When do you prepare suspensions? When there is a insoluble. When the drug is insoluble in the solvent, then you can go for suspension. So depending upon the solubility, dosage form selection is also done. Whether if the drug is highly soluble, then you can go for either tablet or capsule. If the solubility is somewhat less, then you can go for some type of suspensions. Or year of selection is done. Impact stability and efficacy. Solubility governs dosage form selection and drug performance. Suspensions are chosen for poorly soluble drugs like antacids. Which means if the drug is poorly soluble, then you go for suspension. If the drug is highly soluble, then you go for solutions, syrups, injections. Right? So solubility governs, decides, determines which type of dosage form should be selected for a particular drug. Right? Next. Solubility and bioavailability. So solubility and bioavailability both are linked with each other. Right? Bioavailability means the rate and extent of drug absorption from the site of administration to the systemic circulation. Right? Rate and extent. Rate means at what rate? Rate means any process which takes place with respect to time. If you study any process with respect to time, suppose you are moving from one place to the other place, right? So only if you are referred to the distance, then it is distance, that's it. But the distance which you are covering, if you want to refer it along with the time, then distance by time, that becomes speed, which is nothing but rate. So here, how much amount of the drug that is dissolved in your stomach is the amount of the drug, that's it. The amount of the drug that is dissolved. But if you want to analyze, if you want to assess how much amount of the drug is dissolved with respect to time, which means after taking the tablet, after 10 minutes, how much amount is dissolved? After 20 minutes, how much amount is dissolved? After 30 minutes, how much? After 1 hour, how much? So it influences the rate. rate is fast. And extent is also more. Then bioavailability is going to be more. So more amount is present in the blood means immediately that blood, whatever the drug that is present in the blood will reach to the site of action, binds to the receptor, and it is going to show the pharmacological action. So for all these processes to happen, solubility is key. If you can study solubility, you can predict bioavailability. If the bioavailability is good, it is considered that pharmacological action is going to be completed. Right? Which means, see the sequence. If solubility is more, bioavailability is more. If bioavailability is more, therapeutic action is more. Right? So next, low solubility drugs show variable absorption. Right? Variable absorption means what? In stomach more absorption, in intestines less absorption. Right? There are some drugs which are pH independent. So, pH independent means in all pH environment its solubility is not at all affected. Same solubility even if the pH rise or decreased. Right? But weak acids and weak bases, their solubility is going to change. Okay? So in such cases, so drugs show variable absorption. Low solubility, high solubility drugs will never show variable absorption, but low solubility drugs shows variable absorption. Improved solubility improves therapeutic response. So bioavailability depends on dissolution rate of drug. BCS class 2 drugs like ketoconazole. Right? Ketoconazole once again antifungal drug. Right? So here BCS classification system is there, that also I am going to discuss. Biopharmaceutical classification system. So this system is developed based on the two factors which influences the absorption. Okay? Two factors which influences the absorption. Just now I told that solubility is one factor which influences the absorption. The other factor is permeation. Right? Not only solubility is important. Yes, drug is soluble in the stomach fluid, but it has to enter into the membrane barriers present in the body, intestinal membranes. Through the intestinal membranes, it has to travel and then it will enter into the systemic circulation. Which means for that, its permeability is very, very important. Right? Solubility plus permeability. More solubility, more permeability, then you can expect more absorption. Which means, whatever that we are studying right now, right, solubility is going to impact absorption. There is another factor which is also going to impact the absorption, that is called permeability. By combining these two factors, one particular scientist, he has developed this particular amiodarone is a scientist who has developed this particular classification called biopharmaceutical classification. He has classified all the drugs based on their solubility and permeability into 4 categories. There is no other system which can classify the drugs like that particular system. Amiodarone, his classification is perfect. He has classified the entire drugs, whatever the drug that you take, it will come under his classification. Either it is having good solubility or good permeability, or high solubility, high permeability, high solubility, low permeability, high permeability, low solubility, low permeability, low solubility. These are the 4 combinations. 2 by 2 combinations is equal to 4 permutations and combinations. If 2 variables are combined in 2 different ways, then you will get the outcome is 4. Right? So here next example, next slide. BCS classification. This is how BCS classification was developed by amiodarone, who was a scientist, very recently developed classification. And this particular classification classifies the entire drugs into 4 types based on their solubility and permeability. And this is going to predict the bioavailability. BCS class 1 means high solubility, high permeability. Class 2 means low solubility, high permeability. And then class 3 means high solubility, low permeability. Then class 4 means low solubility, low permeability. Right? Now scientists, formulation scientists, for them BCS class 1 drugs are hard cakes. If your drug is highly soluble, highly permeable, then you don't need to make any effort. With a simple formulation of tablet capsule, it can easily absorb into systemic circulation through the intestinal membranes. Okay? But the problematic thing is class 2, class 3, class 4. Class 3, class 4. And class 4 is deadly, very dangerous class. Glycopyrrolate comes under class 4. Right? So here, low solubility, permeability, low permeability. You have to increase its permeability, increase its solubility, then only it becomes absorbed. It becomes, it enters into systemic circulation. Right? So now, class 2 low solubility, high permeability. So here, BCS class 2 drugs we can work rigorously on that. Why? Because just if you increase the solubility, already its permeability more, absorption drastically improves. Right? Next. Here metoprolol class 1, ibuprofen class 2. Metoprolol where it is used? Antihypertensive drug. Metoprolol is antihypertensive drug in the blood pressure regulation. It is used. Then ibuprofen, painkiller, right? NSAID, so that is class 2. Next. Now let us move on to the third one, that is factors affecting solubility. Right? So factors affecting solubility means here physical factors are there, chemical factors are there, environmental factors are there. What are environmental factors? Are temperature and pressure. These are environmental factors. Then physical factors, chemical factors. All the factors which are affecting the solubility can be classified into 3 broad categories. First category is physical factors, second category is chemical factors, third category is environmental factors. Next. So particle size comes under physical factors. Right? So far you know that temperature, pressure, volume of the solvent, pH of the solvent actually responsible for the solubility, contributes for the solubility. Apart from this, size of the particle size of the solute. Particle size of the solute, if it is more, then solubility is less. Particle size more means surface area is less, solubility is less. Right? If particle size decreases, surface area increases. When surface area increases, solubility increases. Right? How particle size reduction leads to increase in the solubility? You tell me. When you reduce the particle size, then surface area increases or decreases? This was the doubt when I was studying in B.Pharm second year. My teachers used to tell me that reduction in the particle size increases the surface area. If you reduce the particle size of the particle reduces, its corresponding surface also reduces. How come this particle surface area increases? That was the question that you are supposed to ask, but you did not ask. I myself will ask the question and I will tell you the answer. Right? Suppose this is one big particle, right? Or else you take one cube. Each side is 1 centimeter. Each side is 3 centimeters, let us say. Each side is 3 centimeters. Now the surface area is 6 a square because it has 6 surfaces, 6 sides, 6 into a squares, which means 6 into a means 6 into 9, 54 centimeter square, its surface area. Right? Now that 3 centimeter cube, if you divide into 1 centimeter, then this side 3 cubes, this side 3 cubes, this side 3 cubes. How many cubes you get? 9 cubes. Right? Each will have 1 centimeter. 6 into 8. 6 into a. 6a square. Right? 6a square means how much? So one side you have 9, another side you have 9, another side you have like this 27 cubes you got. Right? So if you can imagine a cube like this, right? Here when you cut like this, 1 cube, 1 cube, 1 cube, 3. 1 cube, 1 cube, 1 cube, 3. 9. This layer is over. Second layer 9 cubes. Third layer 9 cubes. Total 27 cubes. Right? Each cube is equal to 1 centimeter. Right? 6 into 1 centimeter square. 6 centimeter square. 6 into 27. Calculate how much? 27 into 6. 6 7s are 42. Right? 6 2s are 12. 162 square centimeters. Initial surface area was only 54 centimeters square. But if you divide it into 27 cubes, 3-fold, then its surface area increased by 3-fold. If you add 64 into 3, that number you will get. Right? How much fold you reduce the surface of the particle, that much fold you are going to get the increased surface area. When surface area increases, then what will happen? The solute will get maximum chance to interact with the solvent. And that's how solubility takes place. Right? So here, smaller particle size increases the surface area, collective surface area, not individual surface area. Small particles, individual surface is less only. But when you combine, the collective surface area increases. When they increase the collective surface area, increases the solubility. Right? So next, enhances dissolution rate. Now, question is, what is dissolution and solubility? What is the difference? Right? What is the difference? Dissolution means what? Have you heard of the word dissolution? Dissolution word have you heard? Dissolution, dissolved, dissolution. It means something which is soluble, right? Dissolution means by that word we can guess, we can predict the something which is soluble or something which was dissolved. So the dissolution is also a solubility. But the only difference is, if you measure the solubility with respect to time, then it becomes dissolution. Right? Suppose you are adding solute to the solvent, right? And at every 1 hour, you are taking one sample and analyzing how much amount is dissolved, then it is called dissolution. Solubility of a solute with respect to time is nothing but dissolution rate. Right? Dissolution rate tells about how fast the rapidness, right? The speed of solubility. The speed of solubility. How fast the solubility is. Solubility is fast or slow. That is actually described by dissolution rate. Right? Why dissolution rate is very important in the absorption? Because it influences the onset of action. And in pharmacology, you might have studied about onset of action. Onset of action is how much time the drug has taken to elicit the therapeutic response after the administration of the drug. Suppose you are suffering from some pain. Hereafter, you do this exercise in your home. Whenever you suffer from pain, at the time when you take the tablet, note down the time. Right? And you just observe at what time you got relieved from the pain. And note down that time. And calculate the amount of the time that took for the drug to elicit that particular action. After how much time you felt that your pain was relieved is nothing but onset of action. Suppose immediately after 10 minutes you got relief, then onset of action is 10 minutes. Immediately how it gets relieved? When immediately your drug is absorbed. When immediately your drug is soluble, within short span of time, then it is going to show good onset of action. Right? So dissolution rate is important for that. Next, micronization improves solubility. So when you reduce the particle size, it increases the rate of solubility, it increases the what is called solubility and enhances dissolution rate, and micronization is particle size reduction. Right? So reduction in particle size increases surface area. Example, micronized viscofulvin. So far we have studied that viscofulvin was practically insoluble, but if you can reduce its particle size, then the solubility was increased. People have tried in many ways. Many ways they have increased the temperature, they have varied the atmospheric pressure, they increased the solvent, volume of solvent they increased. They have changed the pH, but still they are unable to increase the solubility. But after reducing its particle size, they observed that its solubility got increased. So it means particle size and surface area, which are linked with each other, influences solubility. Right? Next. Effect of temperature. You know very well, solubility solvent generally increases with temperature. Gas solubility decreases with temperature. Solubility varies with temperature, that is, sugar dissolves more in hot water, which means solids, if you add it to the liquids, right? If you increase the temperature, solid solubility increases. But if you increase the temperature, gaseous solubility in the solvent decreases. Right? So you have in the syllabus solubility of solids in liquids, gases in liquids. Right? In gases, in the case of gases, if you increase the temperature, solubility decreases. In case of solids, if you increase the temperature, solubility increases. Next. pH effect. Weak acids more soluble in high pH. Weak bases, just now I told. So ionization of drug alters solubility. Aspirin more soluble in alkaline pH because aspirin is a weak acid. Next. Nature of solvent. Another factor, that is now physical factors. In the physical factors, important was particle size. Right? Important was particle size. So particle size, if you change, then everything changes. Right? And when it comes to chemical factors, weak bases and weak acid. pH is chemical factor. Another thing, nature of the solvent. Right? Polar solvents, non-polar solvents. You know, if it is a polar solid, then it dissolves in like, it dissolves in like. Polar solids, polar solids. Non-polar solids, non-polar solids. So, solvent polarity affects solubility. Iodine dissolves in chloroform, not in water. So, iodine in molecular form, right? So, it dissolves in chloroform, whose polarity is comparatively lesser than water. Next. Dielectric constant, that is also refers to polarity. So, water has 80, that is polarity of water is expressed in terms of dielectric constant. If you want to measure the polarity of any solvent, the units for measuring the polarity is dielectric constant. If the dielectric constant value is more, then polarity is more. If the dielectric constant value is less, then polarity is less. You can see ethanol, whose polarity is 24, only one hydroxyl group. And if you can see water, where oxygen and two hydrogens are there, its polarity is 80, almost 4-fold increase. 24 into 4, 80. 84, something like that, right? Which means 4-fold increase in the polarity you can see. So reduces electrostatic attraction between ions. Next. Crystal structure. Once again, crystal structure means amorphous solid. Crystal structure means it is once again comes under physical factors. Crystal structure is physical factors. Amorphous form. Amorphous solids, they are rapidly absorbed. Crystalline forms slowly absorbed. Because crystal forms means they are arranged in a proper order. Whereas in amorphous form, there is no order at all. Easily mass transfer will take place. Next. Hydration and solvation. Hydration means the ability of the drug to hold the solvent molecules. Right? If the solvent molecule is water, then it is called hydration. If the solvent molecule is only solvent, then it is called solvation. So hydrogen bond enhances solubility. Compounds which have the ability to form hydrogen bond, their solubility is going to be increased. Next. Complexation. This you might have studied in complexation and protein binding. One of the important applications of complexation is to improve the solubility. So something is not soluble means make a complex, then it becomes its chemical nature is going to be changed, then you can increase the solubility. Next. Co-solvents. Co-solvents means adding another solvent to the existing solvent. Right? Adding another solvent, water is there, and what is highly polar solvent. Suppose you have non-polar solute. Then this non-polar solute actually dissolves in the non-polar solvent. You want to reduce its polarity, then add some co-solvent. That's how its polarity is going to be decreased. Right? So here the co-solvent is propylene glycol, polyethylene glycol, ethanol. Right? We have conducted bike rally. Right? For the bike rally, what was the problem? Diethylene glycol, DEG residues. They have added propylene glycol as a co-solvent in the syrup to increase the stability. Right? But the propylene glycol is having the residues of diethylene glycol. This diethylene glycol is actually responsible for the poor metabolism in the children, infants. Right? In the infants, what problem is diethylene glycol cannot be eliminated by the kidneys. Then nephrotoxicity increased. Kidneys got affected. That's how they have died. Then reduces polarity of solvents. Co-solvents reduce solvent polarity. Diazepam injection using ethanol plus propylene. Diazepam, right? Diazepam solubility is very less, very poor. In such case, what you can do? You can't prepare any other type. So only solution is you should use the solvent, less polar solvent. That is accepted is ethanol plus propylene glycol. In that polarity is less, and diazepam has been dissolved and used as an injection. Next. Surfactants, micellar solubilization. Micellar solubilization you might have studied. What is the micellar? aggregation of monomers of the surfactant molecules, right? So when the surface is saturated with the surfactant monomers, then they have no more space to occupy. Then they rearrange themselves in such a way that they form mesomeres. And in that mesomeres, what will happen? Surfactant molecules will have hydrophilic monomer and hydrophobic tail. And when they reorient themselves in the core, hydrophobic core is going to be present. And lipophilic molecules is going to be absorbed into the hydrophobic core, and that's how solubility is increased. Next. Salt formation is another technique. Right? Salt formation means when weak acid is added to strong base, salt is formed. That's why in your drugs, majority of your drugs, what is their name? Right? Citizen hydrochloride. Only citizen you can't find. Metabolol succinate. Right? So whatever the name that you find on the tablets, its name is always salt form. Why it is in salt form? Because salt form enhances the solubility. Next. Solid dispersion is one carrier will be there, in that solid solution is prepared, and that's how solubility is increased. Next. Hydrotrophy. So use of hydrotropic agents and increases solubility without chemical modification, which means solubility at high concentration. Urea increases solubility of Kf, which means this is a kind of solid dispersion technique. Right? Co-formers. Urea is diffused, dissolved into the Kf, there it forms some hydrogen bonds, as a result solubility increases. Next. So paracetamol moderately soluble, ibuprofen poorly soluble, need enhancement techniques. Next. So formulation relevance is just within 2 slides I am going to complete, right? So formulation relevance means the solubility has tablet dissolution, which means the dissolution of the tablet depends on solubility. Right? More solubility, more dissolution. Less solubility, less dissolution. Suspension stability. If the solubility is more, then what will happen? Crystal growth will take place. Crystal growth is a sign and symptom of the suspension's instability. That is because of the supersaturated solubility of the suspension, because of the temperature variations. Right? That's why storing the suspensions at a definite temperature is very, very important. In the summer season, you have another temperature. In the winter season, you have some more. Because of these temperature variations, there will be some amount of the solid which will be dissolving into the dispersion medium, and when the temperature is decreased, then that will come out as a crystal, and that is going to impact the absorption of the suspension or stability of the suspension. The injectable clarity formulation design depends on solubility. Suspension dosage forms for hydrogen. So analytical importance is dissolution testing, quality control parameters, that is batch to batch consistency. Dissolution testing predicts in vivo performance. Okay? How in vivo performance is predicted by using dissolution? Hour to hour we are going to take the samples. Right? So from 1 hour to 10 hours, which means cumulatively how much amount is dissolved, which means that will serve as an indicator to predict how much amount is going to be absorbed for how much time. That is going to be assessed by using this particular distance. Next. Clinical relevance, dose uniformity. For dose uniformity, drug should have perfect solubility, right? Variable solubility, which means that's what you have said. Apparent solubility. If the drug is having apparent solubility, dose uniformity is not going to be present. Suppose 2 tablets are there. 2 tablets in this, the same amount of the drug is present, but one is active, one is ineffective. Why? Because this is not soluble, this is soluble. Right? So it is solubility which is more important than content uniformity. Right? Next, reduce solubility is critical for drug performance. Multiple factors affect solubility. Enhancements improve, which means this solubility chapter, the knowledge about this particular solubility is very much important in designing and developing a suitable dosage form which can have predictable absorption. You can predict how much amount is going to be absorbed by measuring the solubility and also the dissolution. Right? So how to assess, how to check whether the drug is soluble in a solvent or not? By understanding all these factors. Right? Now, at the end of this lecture, we were able to understand what is that? First, definition of solubility, classification of solubility, factors affecting solubility, then factors affecting solubility, then relevance of the solubility in formulation development also we have discussed. Right? With this, we have concluded our presentation. Thanks for your patience.